Welcome back. Hopefully, your Easter was wonderful. If you celebrated Easter, if not, hopefully you had a good week, good weekend. You celebrated it the way you wanted. If you're wondering what's up with this, you know how I like songs about diseases. Oh, yeah. So this one is rather amusing. It's from an artist, a comedian by the name of Bob Rivers, and he has released many very clever uh, satirical songs. So as always, here's the nice YouTube link if you want to go find this, or just Google Bob Rivers Fluoride. It'll get you there. It's a chuckle. And my goodness, if this winter doesn't end soon, I don't know. What are we going to do? It just doesn't seem like it really is spring, does it? All right. So here we are. We are here. And this is week 12. And I don't know if this joke is too old. Hopefully you know the Roadrunner. The Roadrunner, beep beep. It's an old, cartoon if not. Sometimes I worry that I am getting too old. But I thought this was kind of a cute little nursing joke. It says, everything looks okay. Tubing's not kinked, so why does it keep beeping? Anyway, I thought it was hilarious. I thought I'd share it with you. Now, looking at the syllabus, I realized, oh my goodness, I had too many objectives listed there. We're not going to go that far. We're just going to go here. And still, this is a lot of information, and I know that. Also, as I usually do, I have placed many different quiz questions within the lecture material, and that always makes the lecture longer, and I am sorry about that. So make sure you take plenty of breaks and go get some caffeine or whatever, some protein. Maybe go for a walk and come back. But the good news, of course, is at least you don't have reading assignments, right? So it's all lecture. Otherwise, I could say, well, read the rest in the textbook. Well, okay, you can't. So I have to provide it here with the quiz questions provided as well. All right, so this is the weekly assignment, and this one is rather interesting. I enjoy this video. This this doctor has a very good approach. The title, it's a TED Talks, but from Oslo, and it's why parents fear vaccines. And I like her approach. And of course, like a lot of things, people are very polarized these days. And I was listening to a podcast from my favorite podcaster. He is a professor from Columbia University in New York City, and he's actually a virologist, but he talks about a lot of different things. He does these podcasts called This Week in Microbiology, and they were talking about why people just get so upset about vaccines. And he says, well, and there was one of the professors came on. He says, well, there are people who truly strongly identify with one group or the other. And he said, it's really impossible. You can't get in people's faces. And of course, that doesn't work. That doesn't work because there are intelligent people on both sides of the argument. Probably the best approach, and I totally agree with this, is to appeal to a person's scientific curiosity. As a scientist, that's where I go. Um, the problem is that you have to be willing to sort out a lot of information and get to some get to some really good some facts, some actual statistics, some data, something done from a peer-reviewed scholarly article, for example, and learn about what scientists are doing. Keep in mind, scientists are parents too, and they have wonderful little warm babies that they like to cuddle. And truly, they're not out there to kill babies. Although, my goodness, if you look online, sometimes it seems like they're just evil individuals, and they're not. So, look at what the scientists are saying, not necessarily the manufacturers. You may have doubts about them, but they do have to follow guidelines too, based on what the scientists provide, based on that data, right? So look at this. It's really nice. Of course, it's due by Sunday, 11:59 p.m. So what do you find most interesting about and useful about the video? And then it says, well, and then look at the comments at the bottom. And some are rather shockingly negative. Um, at least they don't say something like you can die screaming, or whatever, and alone. But sometimes they get really aggressive. So, okay, choose one of the comments that is listed, just one, and something that you would like to address. And using information from the lecture coming up, because hopefully I'm going to inform you a lot about vaccines and how they are produced. And no one vaccine is like another. And what is the purpose of a vaccine? Okay. And there are some drawbacks of vaccines. Okay, yeah, sure, they're there. And no label on any drug tells you, oh, it's risk-free. Of course, there are risks. There's risk from aspirin, risk from all kinds of things. But you have to look up scholarly peer-reviewed literature as well. Okay. So you need some peer-reviewed scholarly literature that will actually show you experimental studies. Okay. And I have a good one down here. Actually, it's really good. And I was thinking I might even provide some HPV information for you if you're interested. I might just put that as a supplement to this unit. So anyway, and here is the rubric as always. So, all right. And oh, and here are some terms. Some of these hopefully you remember from some years past when you took anatomy and physiology. If not, well, okay. You will get to learn these, and isn't that's the whole point of this course. So this all has to do with how your body defends itself from the enemy. Now, you've been doing your research now on your pathogen, and you have determined that there are certain features, certain virulence factors that allows your organism to overcome whatever defenses the host might have. It might be that they make toxins, enzymes, sometimes numbers, just by their overwhelming numbers, they overwhelm the host. So, this is going to explain what it is that your pathogen has to get around in order to set up disease. Okay? All right. Even to get in sometimes to cause an infection. All right, so here we have viruses, we have bacteria, and I've only slightly mentioned parasitic worms, but basically anything that tries to get into the body that shouldn't be there is going to be considered the enemy or non-self. Okay? So okay, this isn't exactly what the body does, but sometimes it might feel like the body is sending bombers in and there's explosions of pain. That's inflammation. We're going to talk about how inflammation causes the majority of your symptoms. So if you've been trying to figure out what is it that causes the symptoms associated with my pathogen, well, let's look for inflammation, and inflammation is very much part of the host defense system. Okay. All right. Oh, and one more thing, if you're wondering why this looks like a World War II plane and it looks like a military action, to be honest, it really is. It's them against us. They have their soldiers, little bacterial cells, little viruses, worms, and they all have chemical weapons. So we have our soldiers, or white blood cells, and we have chemical weapons as well. Okay? Okay. And by the way, this is a little bit closer to what our defense system does. So this is a phagocyte, a macrophage, and that's all in red. And you can see it looks very fluffy and beautiful. It turns out the kinds of white blood cells that eat invaders, those are called phagocytes, the white blood cells that eat invaders. And whenever they get triggered, whenever they get excited, they get very fluffy like this. And it's sending its membrane to wrap around the invader, which are these bright green bacteria, E. coli actually. It's kind of a beautiful image. It's a scanning electron micrograph image. Beautiful. By the way, this is a little bit closer to what happens. So here is a phagocyte, a neutrophil, desperately trying to track down probably a Streptococcus pneumonia, couple of cells, and there it just ate it. So yeah, they Track it down. Here's your white blood cell. Again, a neutrophil probably. Here's the bacteria, a diplococcus, probably, again, streptococcus pneumoniae. There it goes. There it goes. It's chasing it. How does it know to find it? It sniffs out chemicals. There's a concentration gradient. The bacterium will send out little chemical signals, believe it or not. Whenever they're swimming, it's kind of like they stink a little bit, and the white blood cell follows the trail. Kind of cool. So we'll take a moment just to appreciate how wonderful our immune cells are, and you better treat them right. I'll tell you how later. Let's start with the basics. Where and what, I don't know why that's capitalized, is involved in host defenses? All right, so when breaking this down, most immunology textbooks and immunology professors kind of break this down into three phases or three lines of defense. And like I said, it is a military action. We're going to go over everything in more detail, but this just breaks it down. Now, by the way, how resistant you are to infection and disease depends on these three major methods of defense, these three lines of defense. The first line of defense is the barrier. Okay, so I have the Great Wall of China. Truly, quite the barrier. And it's going to block pretty much everything. So this is the barriers in your body are innate. You're born with them. Innate means they come with you. They don't, you just get them when you're born. They are, and it's not going to get any better with time. You've got skin, it blocks invaders, and you got it throughout your life. You're born with it, okay? And a lot of times the barriers will slough off, but then it will be replaced with fresh barrier material. So every once in a while, the wall needs to be fixed, and then you just bring in more bricks. It is non-specific. I mean, this wall would stop deer as well as enemy invaders, which is what the Chinese were intending. So it's very non-specific. So it's just designed to block anything that shouldn't get in, period. Okay. Now understand that a barrier, the first line, is really not part of immunity. Immunity is something that gets learned. It gets better with every, it's kind of like you exercise, it gets better, it gets stronger, it gets faster. All right, so now the second line, and we'll find the second and third lines are involved in immunity. Okay. Now, the second line, for the most part, is also innate, but there are parts that develop over time. So you are born with this. And again, it's mostly non-specific, but every once in a while they get a little picky about who they take out. So our soldiers are not going after deer on enemy territory. No, they're going up, they're going to find the enemy. And a lot of times they blow up like cells that the enemy might be hiding in. The third line is more like James James Bond. Very highly trained and adaptive. So it learns, it gets better, it gets faster. Okay? Stronger with each exposure. Very specific. James Bond, you don't see him marching with the troops. No, no, no. He's very, very specific. Actually, specificity, we're going to find out, has to do with the cells and their receptors. The receptors have to specifically identify something as foreign. Okay, so it only goes after what it is trained to go after. Again, it's all about receptors. Now, mind you, the third line, okay, is going to be some kind of interaction between self, the good guy, and non-self, somebody that doesn't belong there. And understand they all do work together. Sorry, with this new slide format that I'm using, it got cut off a little bit there. Sorry. So, what are we trying to keep out? Okay, let's look at the barriers. Okay, so what are we trying to keep out here with our defenses? Okay, so it's time to put a name to it. We have to put a name to the invader. We're calling the invader something that doesn't belong, something non-self, as an antigen. That's what immunologists call it. Okay, the invaders are called antigens. Antigens are anything that generates an immune response. Now, where did the name come from? Well, we used to say and define antigens as something that generates the production of protective host antibodies. So it would generate antibodies until we found out that the immune response actually does so much more and makes so many more things than just antibodies. Okay, so it responds in many other ways. So now we just say antigens generate an immune response. Okay, now, sometimes the antigen is something really, really big and complex, and sometimes it isn't. So it could be a parasitic worm, bacteria, bacterial toxins. So here's our bacterial cell over here. Whoopsie. And a little fragment came off. That's supposed to be his bacterial toxin, an exotoxin released, okay? So it could be viruses, pollen, whatever. Complex antigens usually contain lots of different identification tags on them. So the whole bacterial cell in itself, by definition, is an antigen. Yet it has many different kinds of ID tags kind of sticking out of it. And you can see these little things sticking out of it. Okay, well, to be honest, they're all pili, but okay. And flagella. But okay, imagine these as little chemicals that are ID tags, and they're on flagella, capsules, if it has a capsule, bits of cell wall material, all kinds of things. Okay? All right. So sometimes we want to define the exact structure on the antigen that is being identified. We only have to do this if the antigen is big and complex, okay? If it's very small, if it's just a little tiny piece of protein, then we don't have to do this really. But okay, if an antigen has a lot of ID tags, like I said, then we call each type of ID tag an epitope. Here we have a cartoon illustration of one big bacterial cell, supposedly antigen, but yet it has three very unique epitopes ID tags embedded, okay? So which ID tag epitope would produce the strongest immune response? With Mr. Seberg, you learn that there's lots of different organic molecules that a cell might have. So what might be sticking out of a flagellum or a cell wall or a cell, membrane or a capsule? Well, of there's lots and lots of proteins that do stick out, and really proteins make the best epitope. The proteins are going to give you the strongest immune response every time. That would be like your exotoxins, the things excreted from like Clostridium difficile, Clostridium botulinum, Clostridium perfringens, Staphylococcus aureus, Streptococcus pneumoniae, Streptococcus pyogenes, all those things, okay? So those are really, really good. Now, the lipids are not as good. So when we're looking at gram-negative ID tags like LPS, endotoxins, it's not as good. So if we're looking at, the LPS structure of Helicobacter pylori or E. coli or Salmonella or Pseudomonas or Neisseria meningitidis. It's not so good. The worst of the three are the carbohydrates. Carbohydrates are not really seen very well at all by the immune response, even later on in life. When you're young, when you're a baby, yeah, you're not going to see these at all. Later on in life, even so, it's just not processed very well. Carbs make really horrible antigen epitopes. They don't really generate an immune response. So let's say this bacterial cell was covered in a glycocalyx, a sugar coat, right? Glycocalyx is a sugar coat, so that's a glycocalyx capsule. So how would your immune response respond to that? The answer, as adults, not very well. As children, probably not at all. Okay. So yeah, children really don't respond very well at all, and that can be scary. And this is just an image. This is Neisseria meningitidis grown on blood agar. And you can't really see the bacterial cells. They're kind of hazy. But that clear zone is the part that isn't being, or sorry, this isn't on a, sorry, this is Neisseria meningitidis grown on, or viewed under a microscope. So they're making capsules. Sorry. Okay, so Question. This bacterial cell, once inside of a host, generates an immune response. It is called an epitope or an antigen. Come back. Okay, so the bacterial cell gets inside the host, generates an immune response. Okay, so what are we talking about? By definition, it is an antigen. Okay, if I asked, okay, what is the specific part of the bacterial cell that is identified by the immune response? That would be the epitope, the little ID tags there. Okay, how many epitopes would be found on this bacterial cell? 1, 2, numerous. Yeah, the antigen, this bacterial cell has numerous epitopes. Okay. All right, so let's talk about the first line of defense, the innate barrier. So here we have a model of what, basically our first line of defense. So we're going to go through these one at a time. First, understand microbes love you. You're warm. They love them, especially the moist areas. There's probably a lot of food there, very nutritious. But yeah, they really prefer the moist areas the best. Dry skin, not so much, and we'll talk about it. So they're going to try to get in where there isn't a barrier, and they will in every orifice they can. And we do have ways to stop them. And that's what we're going to talk about here. So number one is the skin, our first line of defense. Treat your skin very well. And actually, your epithelial cells in general, they are your protective barriers. And this is just showing you again the layers of your skin or the layers of your tissue with the outer skin, the epidermis out here. And out here you have keratin, which is that hard protein substance, which is like scale material, very protective. And it'll slough off. So if any bacteria, try to hold on, well, they just fall off with the skin cells, which is why so many of your skin cells end up as house dust. Oh, yeah. By the way, yes, that sloughing off is also what happens in the GI tract. Lots of your old cells come out in the feces along with the microbes. So it's a replenishment. Every time this gets replaced, the cells fall off and it takes the bacteria with them. Okay, and by the way, your skin has a pretty low pH. It's acidic, and microbes are not very tolerant of acidic environments. There are exceptions to this, by the way. Fungi love acid, so there you go. Perspiration, it turns out that perspiration uh, not only cools you, but also there's a chemical in perspiration, an enzyme called lysozyme. Lysozyme is a digestive enzyme our cells produce to break down peptidoglycan cell walls of bacteria. So the bacteria try to hang out, and we release lysozyme in the perspiration. It eats them up. That's pretty cool. Okay, so this question asks, so how do the microbes get through? Well, they get in when this barrier, the skin barrier, is breached. So when you're in surgery, when you're putting in any kind of an IV or an injection, or when they have a wound like a burn wound or anything like that, an abrasion or a lesion. Okay, could be even a lesion due to an infectious organism. So, yeah, the barriers are breached. These areas are extremely vulnerable. So, you have to be very careful. Okay. So another barrier, tears. Like I said, the microbes, when they can't get through the skin, some of them try to get through your eyes because it's moist, it's warm, and it's an opening. But you have tears, okay? And here, okay, so you get this washing whenever you have tears, and it's hard to see the tears coming down, but tears will literally wash the eyes. And you also have mucus in the eyes, a type of mucus, and it will capture a lot of the microorganisms there. So that tearing action is very good. Okay, so it washes the eyes. And guess what? Tears contain that enzyme known as lysozyme. So if you have any bacteria there, hopefully the lysozyme will break it down. So how do they get through this barrier? Well, okay, they get there by this method. And okay, some of them are very virulent. They can manage to get through anyway. But when you rub, sometimes you break down skin that is there. So don't do that. Get a Kleenex. If you don't have a Kleenex, use the inside of your shirt or something like that, but don't rub your eyes. I know people will wonder, what's wrong with you? Are you okay? And you just have to tell them, no, my eyes itch and I don't put my hands on my in my eyes. So don't want to inoculate my eyes. Thank you. Okay, so what's another first line barrier? Well, like I said, sometimes the microorganisms, they want to get in places that it's moist, it's warm, there's a lot of food, and the best place to do that is through the mouth, the oral cavity, right? Well, then they have to get through the stomach to do that. Okay. One of the things I didn't mention too is that your mouth has a lot of saliva, and saliva contains lysozyme, and it washes your teeth and gums. But let's say they get past that and now they're traveling through the stomach. Okay, turns out the stomach is very acidic. They would have to have huge numbers usually to get through the stomach acids. They would have to. Okay. And let me just tell you though, one thing you should know. You are always taking in bacteria and viruses and other things with every mouthful. Every time you swallow, whether you're eating, drinking, or just swallowing, you are bringing in bacteria. But the acids in the stomach usually take care of them. Okay? All right. So there's also acid in the vaginal epithelium, ladies. So in the vaginal area, you have a lot of normal microbiota hanging out there, and you are actually feeding them. So these are good bacteria, and they're happy. And you do feed them, but only when you're making enough glycogen, and that's during your reproductive years. But they're there, they're happy little bacteria, and they're making a lot of acids. And again, a lot of microorganisms aren't going to like that. So how are the microorganisms going to get through? So, all right, you have all that stomach acid. Well, how are they going to get through that? Well, number one, sometimes if you ingest huge amounts of bacteria, they will get through and cause an infection and a disease in the intestines. Plain and simple. And that's what they want. Fecal oral contaminants want to get to the intestines. Okay. But the stomach would stop them unless you do something silly like maybe take antacids while traveling or some other time. Antacids are just not a good thing unless you absolutely have to take them. So here's a scenario. Let's say you're out traveling and you've been a little careless and you've been eating food not fully cooked or fresh fruits or vegetables because they look lovely, water that isn't bottled. You're doing kind of silly things. And then to, kind of top it all off, you go out drinking and you're not feeling so good. So then you have to take an antacid. Okay, you're not a silly person, but maybe you have some silly behavior here. So you have just neutralized your acid and then you're consuming lots of bacteria and you have no barrier there to stop them. So when traveling, don't take antacids and don't drink. Again, eat the foods that are fully cooked, okay? Bottled water, and maybe drink Coca-Cola or tea or coffee or acidic beverages. Okay. Other things. How about down here? How do we lose our acids in the vaginal area? Well, again, our little protective guys, our normal microbiota, they are here and you killed them with an antibiotic. Dang. By the way, what else can happen? Well, as you get older, your hormones change. Well, if you're very young or very old, you're not making enough glycogen to feed your normal microbiota. So unfortunately, yeah, as we age, we lose our normal microbiota, we lose our acids, and we are more prone to infections. Okay, vaginal infections, yeast infections, urinary tract infections in general. Bummer. Ladies, we can look forward to this later, right? All right, so what's another barrier? Okay, mucus. Oh, poor little thing. Mucus is this gooey, sticky stuff, right? And it works like fly paper. It traps microbes, though, and it traps them in the respiratory tract, the genital tract, the GI tract. Again, you kind of even have some around your eyes. I mean, it's everywhere. You get it everywhere. And then, okay, if it's in the respiratory tract, one of the things that you do are you have these little cilia, and they will beat and they will carry all the little particles, bacteria, viruses, or whatever, up, and then it gets in the nasopharynx, and you either swallow it, let the acids take care of it, or you spit it out. So, all right. Um, Again, cilia are very important in the respiratory tract to help with that mucus, or to help move the mucus and get them, up and out of there. Cilia is good. By the way, mucus contains lysozyme. Yes, it does. Okay, so how are the microorganisms getting past the mucus barrier? Okay, well, we did talk about some viruses that make neuraminidase. That is one way, and I'm not going to put that again in here. That would be influenza does that. But when people smoke, that damages the cilia and the mucus and the microbes, and everybody just kind of stays in the lungs. People, when they have cystic fibrosis, they have such thick mucus, they can't get it out. And so the microbes get stuck, in the small little areas as well. So, okay. So smoking, sorry, that should have come up a little sooner. Um, and by the way, microorganisms, a lot of bacteria like Bordetella pertussis, very good at making toxins that destroy cilia. This is a very famous picture. So these would be respiratory epithelial cells, and they're supposed to all be covered in cilia. If you look very closely, there are these tiny little rod-shaped bacteria. Okay, where are the bacteria? Little arrows are pointing. But they produce toxins that just clear cut the cilia. A lot of bacteria do that, but Bordetella pertussis is very good at making this toxin. If you're wondering, I've never heard of Bordetella pertussis. Oh, pertussis maybe in like a DPT shot, but okay, think whooping cough. There's that horrible commercial. I hate that. It's terrible. It freaks me out. I want to go to the kids' rescue. Anyway, speaking of which, severe and prolonged coughing can damage also tissue. So it can damage everything. And that's another way for microbes to get through. Okay. So we're going to talk about an inflammatory response, and you get all kinds of damage, and that'll allow the microbes to get through. Oh, sorry, this came up a little faster, but how about in the urinary tract, like the urethra and all of that? So, okay, so urine flow actually does flush the urethra. So every time you urinate, you are flushing all the little microorganisms that are trying to get in through the urethra. So good flushing action is very healthy. So you drink a lot of water, right? And that's why also when you're getting a urine sample, you take the second stream because you would have flushed out probably the majority of the little microorganisms that are hanging out, at least the ones you didn't wipe with the little antimicrobial agent. So how are the microorganisms getting through this barrier? Well, a lot of microbacteria swim. They have flagella. Some of them, once they get in, they hold on with pili, right? We talked about that. And also we insert catheters. And, oh, sorry, sorry, we insert catheters. And that is good. Now, this is a model, but all of the little microbiota that is right at the edge of the urethra, okay, once you insert this long tube, you are going to shove microorganisms into the urethra. You are. Same thing kind of happens with tampons, ladies. Hate to tell you. Okay. How about another line of defense? Normal microbiota in general. So here we have all kinds of Lactobacillus acidophilus. Aren't they nice? And what is this little pink cell that we've got here? This is a vaginal epithelial cell. Here's the nucleus, and it's even pink. Just and no, that's just the way it's stained. This is a gram stain. Okay, so they stained and they're purple, the bacteria. So they are there and they are making acids, and a lot of times they're making other antimicrobial agents. Sometimes they're eating all the food there. They're trying to keep the bad guys out. So good normal microbiota and just not in the vagina, okay? Not just there, but throughout your entire body. So we should treat them well and only take antibiotics when necessary. We don't want these guys hurt. By the way, in addition to antibiotics changing your normal microbiota, any change in diet will do this. Change in altitude. Yeah, you move to or from Denver. And again, it turns out that the bad guys, they move in and they tend to be able to handle these changes a little better than the normal microbiota. So, yeah. How about a question? What is the name of the enzyme that breaks down bacterial cell wall peptidoglycan and is found in most all body secretions? Tears, saliva, mucus, perspiration. Is it penicillinase, lysosome, lysozyme, protease, peptidase? You said lysozyme. You are right. C is correct. Good job. Okay. Okay. How about another first line defense? How about chemical weapons? Well, I'll let you know that back in the day when I studied microbiology and immunology, we really didn't know a lot about these chemical weapons. I'm only going to just give you a general sense of what the body does send out to destroy these microorganisms. And especially, we know a lot about the types of chemicals that destroy bacteria and cellular creatures. Not so much, we're working on the viruses and other things, okay? But okay, one of the chemical weapons, we use a lot of oxidizers, some of them very close to hydrogen peroxide. So, they form ions, and the ions are charged and they go around and they beat up on cell structures. So, that works pretty well. And then there's a huge group of protein molecules called defensins, and they will form little pores in the bacterial cell. So the little gray little, thank you for, I hope you like my animation. So this is the price to pay when we don't buy a textbook. You have to put up with my animation. But they're little pores, little chemicals, and once they punch holes in the membranes, well, then everything leaks out and the bacterial cell, well, it doesn't survive. So very nice, defensins. So why don't these chemical weapons always work in the first line of defense? Well, a lot of them work on some invaders, but not all. So again, this would work on a bacterial cell because they have membranes that are semi-permeable, and that's what they're supposed to do. But viruses, no, it really wouldn't work on those. So yeah, that's one of the reasons. Okay, moving on. Let's go into the second line of defense, and this includes innate and adaptive immunity. What's innate? Born with it. Adaptive, you learn, get better. Okay, so before we go into this, because this is huge and it's very difficult to teach the subject because everything interacts with everything else at one time. Okay. And so the best way I can do this is just break it down into little bits. I'll give you the where, what, and how, and then sort of put them all together a little bit later on. So, and even before that, let's define some terms. Remember, the second and third lines are involved in immunity. The first line, the barriers, are not. They're just the first line of defense. So true immunity is the second and third line of defense. So maybe we should define immunity. Yeah, let's do that. Okay, so immunity is the study of the body's second and third lines of defense. What? Okay, so it functions. Here are the functions, and this is the way it should work in a healthy immune system. Number one, the immune system should do surveillance, checking it out, traveling throughout the body, and they do. Immune cells go everywhere, except there are some places that have barriers. There's the blood-brain barrier. the testes, and the eye humors. Those are, I think, the only three places where the immune cells and response doesn't go, they don't go there. Okay. The second, okay, function of a healthy immune system is to recognize the foreign material, identify them as an antigen. Okay. So they have to go around, travel throughout the whole body, checking non-self ID tags, looking at the antigen or the specific epitope sticking out of the antigen if it's big. Okay. Number three, okay, you got to destroy it. Destroy non-self invaders. Those are the three functions. Makes sense. So we need to look at the major players that are involved in the immune response. Okay, so what are we talking about here? What is this including? Well, the big players would be the blood cells, and there are several. So here's some a single white and some red ones here, and oh, that's an invading virus. It's kind of cute. Okay, so the red blood cells, they really are important as well because the red blood cells or erythrocytes are there to carry oxygen to all of the tissue. And what does that have to do with the immune response? Well, let me tell you. This little soldier here, the white blood cell, he needs to make energy. And he can't do that without oxygen. And who brings the oxygen? The red blood cells. So the red blood cells have to carry oxygen to the fighting white blood cells if they are to make that cell energy, or what we call ATP. So if you're anemic, yeah, your white blood cells aren't getting enough oxygen, so they're not getting enough energy, ATP. Not good. Now, there's also the white blood cells. In addition to the red blood cells, there are the white blood cells. Another name for white blood cell is leukocyte. Cyte, always cell. Leuc always means white. Okay? These are the soldiers. These are the guys, the military personnel. And now some of these white blood cells make protective antibodies. Okay, we'll talk about that a little bit later on. There's also some other components of the blood, okay, that aren't exactly cells, but we'll put them in there. So these are the platelets, and they do help to repair any damage. If there was an assault, collateral damage after the battle, the platelets will come in there and they will help, and they will make sure you don't bleed too much. Okay. That's not good. There are also, in addition to blood cells, there are second line defense chemical weapons and chemical signals as well. Okay, so these are different than the chemical weapons that we learned about before. These are different. Will we talk about them? Yes. The big one, complement. And complement has a lot of functions. Okay. And the chemical signals, something called cytokines. Don't worry, we're going to talk about these each individually. Where are all of these blood cells made, you ask? I'm so glad you asked. Okay, so it's all in the bone marrow. Think about all your bones. It's not just there to keep you upright. There's so much going on in this tissue. It's amazing. So blood cells are made in the bone marrow. You have all of these, what are called progenitor cells. Sometimes we call them stem cells. And they will make all the different kinds of cells. So here we have the bone marrow. And this is just a close-up view of some of the cells, making more cells and making more cells. And this just shows how they differentiate or become the different lines of blood cells. And there are many. Now, the making of blood cells is called hematopoiesis. Now, sometimes, when the body needs more blood cells, there are chemical signals out there called colony stimulating factors. You don't need to know that, but if it ever comes up in your clinical life, you'll know what that is. Okay, so all of the blood cells, they're all pretty much going to be found in normal blood. Maybe not in huge numbers, but you are going to find blood cells everywhere. Now, certain numbers definitely increase during infections, and a lot of times we can look at a blood differential, and see, okay, you have a lot of white blood cells, leukocytes. Okay, there's an infection going on there. If we see eosinophils, we'll talk about those. Those are special kinds of white blood cells. We'd say, oh, you either have allergies or a parasite. More on that later on. And these blood cells, they move throughout the whole body, traveling through circulatory systems. Certain kinds go to various tissues. And this is just to kind of show you, again, breaking it down. This is kind of cute. I found this online and it just shows hematopoiesis and what happens. So you have a lot of hematopoiesis going on in a prenatal, early in life and then postnatal. It's kind of interesting. Yeah. All right, let's talk about the different types of cells. And this just shows how you start off from a stem cell in the bone marrow, okay, which is a stem cell, okay. And if it divides in one direction, well, then you get your platelets or white blood cells, or you get your what are called granulocytes. But if they go a different direction, then they make the agranulocytes, the ones that don't have granules, really. And that includes something called a natural killer cell and lymphocytes. So we'll get into that in just a moment. So let's talk about the four types of leukocytes and begin by looking at the granulocytes. So called because the granulocytes keep their chemical weapons in granules. What are the chemical weapons? Well, one of the big chemical weapons is histamine, but there's a lot of other inflammatory chemicals. So here we have here are the three big granulocytes here. The basophils, it doesn't show, and well, the mast cell also is included in that, and the eosinophil and a little bit of the neutrophil. So the basophils, again, this guy here, they circulate, and you see these in allergic reactions. Actually, you see these in all allergic reactions. The mast cells, you see these more in the tissue. But again, yeah, you'll see them during inflammatory responses and allergies. Eosinophils, this guy here, you're going to see these when there's a parasite or allergies. So when you see a lot of these different kinds of cells, you go, oh yeah, allergy, oh, parasite. So yeah, okay. And then there's another type of granulocyte. This is the neutrophil. And this little guy is here. You got to love your neutrophils. They are made in the highest numbers. So when there's an infection, you usually get a lot of these neutrophils. And they don't live long, but they get there. Okay, and their job is to engulf and destroy bacteria. By the way, when these cells attack, they don't survive. They basically explode, releasing all of their granules. And let me tell you, the contents in those granules kill them and pretty much everything around them. So, yeah, so they don't live. And again, the neutrophils are very short-lived. Okay, now let's look at another type of white blood cell. These are the mononuclear phagocytes or monocytes. Here they are. And the monocytes, here's a nice one. You can see he's busy. So the monocytes circulate in the blood. A lot of them develop into macrophages like this guy. And macrophages are huge eaters. Phagocyte literally means, big eating cell. Okay. And the cell, this is all the cell, itself, and it's eating these little particles here. That's what the circle is, and it's bringing it into the cell. It's eating them. That's phagocytosis. And yeah, the macrophages are like the big garbage cans of all of your cells. They go around, they eat pretty much anything that doesn't belong there. They just have to identify it as foreign, and they also have to grab onto it. Sometimes that's not easy. So it'll grab invaders and cell debris after a battle, just pretty much anything. Okay? All right, another type of cell, the dendritic cells. All right, so dendritic cells go everywhere. This is a cartoon of a dendritic cell. This is a really important guy. He does so much. They do go everywhere, but they really like to hang out in lymph nodes and lymph material. I'll talk about that a little bit later on. So his job is to engulf material as well, but he not only eats it, like here's a virus, Okay, I had to shrink it there, and he brings it in by phagocytosis. He breaks it down by actually some of the lysozyme enzymes, but then not just that, they will present it out on their surface, and they will present it to the other white blood cells, the other military, as if to say, okay, I saw something that looked like this. Are you designed to take care of this problem? If so, you and I, we need to talk. We need a handshake. We need to talk, and you need to go after this. We need to start a war. Okay? So we say that the dendritic cell is an antigen presenting cell because it engulfs material, presents antigens, and again, it does this not only for this part of the immune system, but also um, it's important for the adaptive immune response. Okay, so these phagocytes are antigen presenting cells. They're not the only ones, but they're the biggies. So here we have this is a T lymphocyte. Sometimes we just want to say T cell. It's easier, but it's the same thing. If it's a cell and there's a T there, okay. So this dendritic cell had to go find the right T cell, and you can see they're having a handshake. They need to check each other's ID tag. They need to, again, there's a shake here and a presentation there. So, and then there's going to be chemical signals going on. They have to talk to each other. So the dendritic cell is the one that tells this military leader, the T cell, we start a war. And the enemy looks like this. I spotted it, so now it's up to you to go get it. Okay, the T cell then becomes activated. Okay, and again, war. Okay, so when it presents the antigen to the T lymphocyte or T cell, it activates the T cell to start a war. Okay, the fourth kind of white blood cell are our lymphocytes. They are mostly going to be talked about in the third line of defense, the adaptive. Okay, and again, they're going to be the ones the T lymphocyte's going to be talking to the dendritic cell. All right. So again, they're responsible for learning, adaptive immunity. They get smarter and smarter. So this includes B cells and T cells. B lymphocytes or T lymphocytes, same thing. They're very highly specific in recognizing a particular type of antigen. So they don't go after everything, just what they've been trained to do. Generally, they hide out in lymph nodes and specific lymphatic material, especially in your, as it turns out, your gut epithelial cells. Another lymphocyte very closely related to some T cells is the natural killer cell, and he goes out and he destroys certain types of cells. We'll talk about that a little bit. Time for a quiz. Which of the following cell types carries chemical weapons inside granules and is considered a granulocyte? Basophil, macrophage, lymphocyte, monocyte, erythrocyte. That would be the basophil, or eosinophil, or mast cell, or neutrophil. All of those. Okay, basophil, mast cell, neutrophil, or eosinophil. They're all packing granules. That's their weapon. All right. So let's take a look at where they're going. All of these cells are circulating throughout the lymphatic system. Again, some have preferences. They like certain areas better than other areas. Again, it's rare that they can't get to an area. So they can't cross the blood-brain barrier. They can't get into the humors of the eye, and they can't get into the testes, which I can't point out in this figure, but you got that. So how do these immune cells know what to do and where to go? Did you ever ask that question? Oh, so there's an invasion. There's something going on, but how do they know? How do they know where the battle is and how to get there? Well, that takes cell communication. And there's lots of ways to communicate. Okay? But communication, like with every aspect of this world, allows a coordinated and effective response. Okay, the first one I'm going to talk about are these adhesion molecules. Let's say the battle, there's an invader going on right here. These epithelial cells will in fact recognize that something's going on, and they will shoot out these little receptors, little adhesion molecules. Now the white blood cells are roaring past 100 miles an hour. No, well, not that quick. but pretty fast. So they go whipping on by if they didn't get stuck on these adhesion molecules. And also, as they approach, these cells also knowing that there's an invader, they get a little leaky. So the white blood cells can leak on through and get to the battle. Okay? So that's what endothelial cells tend to do. So these would be endothelial cells of the blood. Okay, they line the bloodstream, blood vessels, and endothelial cells adhere to the phagocyte white blood cells, allowing them to exit the bloodstream at the battle location. And this just shows you a really cool illustration. So they're whipping through here, but you can see the white blood cells are sticking. Okay. And not just that. Okay, eventually they'll get the right adhesion molecule and they slip on through. Endothelial cells are very cool. All right. All right, how about some other communication? Well, all right, so you have the battle on one side and the military force, the white blood cells on the other. How do they know where to go? Well, okay, there are chemical signals known as cytokines, and they include a huge group of chemical signals, and they are produced by many different kinds of cells during damage. If there's like damage, an invasion, an attack, anything. So cells throughout your body send these things out. They're a lot like hormones. Okay. If you remember, that's how all of the cells kind of coordinate in your body. So, the uterine lining cells know to start to contract when there's a baby, right? When there's labor, when labor needs to happen. So they're a lot like hormones, again, produced by many different kinds of cells. And certain cytokines have certain jobs, and they attach to certain cells with certain receptors, and they cause the cells to change. So here's a white blood cell, and let's say if the signal isn't meant for him, he ignores it. But if the signal is just right, it attaches to his receptor, and then he is activated and he grows and he divides and makes lots and lots of baby cells, and they differentiate into different kinds of cells to do different things. It causes cells to move. And some cells also, if they get a cytokine, it might tell them that they themselves are the enemy. They've become let's just say they're possessed and they must then die. Okay. A lot of intracellular pathogens get in and they get into your own cells. We talked about viruses, they become part of you. So if you're an infected cell and you get a cytokine, it might tell you you have to kill yourself now. So that's something called apoptosis. Types of cytokines, again, there's so many. I'm even going to leave two major types out. There are cytokines called the colony stimulating factors, lots of those, CSFs. They will tell a lot of white blood cells to multiply, to differentiate, so they're better soldiers. There's tumor necrosis factor, again, a chemical signal that draws in more white blood cells for inflammation. Also, this is a chemical that tells cells if they've been invaded to kill themselves. So that's the cell suicide chemical signal, or that causes cells to die. That's called apoptosis. Somebody tried to make a lot of, if you're wondering about the name, the founder, the person who invented this or discovered it, thought this would be a big money maker because he found that it did in fact destroy tumors. But you inject this into a person, it's so toxic, it's very bad. So, but you do need to know interferons, and by golly, you already know this one. Remember, interferons, they interfere with viral replication in viral infections, and they will regulate the inflammatory response, sometimes causing a huge inflammatory response, which is why some interferon therapies are very toxic. There's also, sorry, didn't mean for those to come up. There's also interleukins. So looking at the name interleukin, that would be in between white, in between the white blood cells. Yeah. So that's a signal between white blood cells it's kind of like when military personnel have their own channels, right? Their own frequencies that they talk on. Yeah. Understand all the cytokines work together to promote a response. Also know that any overstimulation of cytokines can cause what's called a cytokine storm. So maybe you have one little infection causing disease there, but because you really start to cause a huge response, you get symptoms throughout the whole body. Now, again, it can mask your original source, but it can also be deadly. What can cause a cytokine storm, you ask? Remember the super antigens? Remember, like staph and strep, they make super antigens that just stimulate too much of the white blood cell uh, uh, activity. So yeah, those exotoxins can, super antigens can do that and kill you. Okay, there are also cytokines that say, let's have a fever. Oh, joy. A fever is actually an important host defense mechanism and a strong indicator of an infectious disease versus something else. Not 100%, but there you go. The chemical signals that produce uh, fever and therefore are cytokines, these are called pyrogens. So, pyrogens are cytokines produced by macrophages and microbes and a lot of other different cells. So these chemical signals, the cytokines, they have a target. Where are they targeting? Well, it's going to be the hypothalamus, right? If you remember your anatomy, it's the hypothalamus in the brain that regulates body temperature. So it tells the hypothalamus, raise the body temperature. Why would it do a thing like that? Well, so with a temperature rise, as long as it's not too high, it increases how the enzymes work in the cells, so cells are more efficient. It will enhance the inflammatory response. It enhances phagocytic killing, the num num num, the white blood cells eating the bacteria and other things. High temperatures will trigger multiplication of the white blood cells, so the lymphocytes and the neutrophils. And it also will cause a release of the white blood cells from the bone marrow. Few other things. It also will help the body tie up iron and zinc and keep it away from the bacteria who would really like that. So, why do we try to bring a fever down? Well, in little children and in older adults, fevers can be very devastating. So you never want a fever, especially a high fever, in a little one or in an adult or an elderly person or in somebody who has a serious illness. But if you are relatively healthy, okay, relatively healthy, and up until the point where you got this infection, all right, and you have a fever, if you let the fever run its course and you don't take the Tylenol, you will actually get over it quicker. So let's say you get sick the week before finals. You do not want to be sick during finals week and you have a bit of a fever, maybe let it run its course. But if you have the fever during finals, okay, you can't deal with it. So you just got to bring that fever down to feel better. Or maybe you need to stay home. That could be bad too. Maybe you don't want to be out school or work, right? If you have a fever. All right. This is something very interesting. In 1934, Wagner Jauregg he had suggested a transfusion from a malaria patient to a neurosyphilis patient. So one patient had malaria and he's giving the patient with syphilis malaria. Why would he do that? One of the major symptoms of malaria is fever. And because syphilis was so deadly and we didn't have any treatments that pretty much wouldn't kill the host, this was to induce fever to treat the syphilis because that was much worse. Hoping the malaria this gentleman had was not one of the nastier kinds. We'll assume that. I know, freaky, right? Something to talk about over Thanksgiving dinner. Hey. All right, another chemical that your body makes. Now, these are actually protein chemicals, and they are freaky, called the complement system. Complement complements the adaptive immune system. So even though they're part of the second immune response, they're really important. They're going to act and help out the entire adaptive immune response, not only in the second, but also the third line of defense. So these are freaky, versatile little proteins found throughout the blood and tissue. and they behave very unusually. So they split into fragments, and for example, there's a C3 part of the complement. It splits into a C3A, and the C3A goes away, and the C3B binds to the target cell. So here we have some C3Bs attaching to the bacterial cell that's about ready to snuff it. Okay, there are three functions of the complement system, and you don't have to learn the complement cascade from me. If you ever have to learn the complement cascade, let's say you take an immunology class, you can give me a call. I will help you learn this. It's very complex and even drives my pharmacy students crazy, but you do need to know what it does. So, its function. Number one, it forms a membrane attack complex. So they break down, they rearrange, and they form pores in the bacterial cell. And we sort of saw that before, didn't we? Yeah, with defensins. Okay, so once you punch a hole in the bacterial cell, the cell will lyse. And yeah, this is really probably only going to work in gram-negative bacteria, though. Okay. It only really works on the outer membrane of gram negatives. Okay. Another thing that it does is opsonization or opsonization. I know Gesundheit means to make tasty, to opsonize. This is a complement protein part that becomes very, very sticky and will attach again to bacterial cells, viruses, or other things that don't belong there. And they stick to it. Why? Because then a big phagocyte, like a macrophage that's very hungry, maybe it wouldn't be able to hold on and grab onto the red bacterial cell very well. So complement attaches to something specific on the bacterial cell and it makes it all sticky. The blue part's very, very sticky. And then the phagocyte can attach to the complement. And when it brings in the complement, it brings in the whole red blood cell. So they all come in. Okay. So it just means to make tasty so phagocytes can grab on and eat them, eat the invaders. Okay. So it's a little piece of sticky protein. Helps phagocytes grab invaders. The last part, they do some of the protein parts fragment in the complement system, and they can cause a huge inflammatory response. So it works just like a signal, kind of like the cytokine, but we don't consider it a cytokine really per se. But it does attract a lot of the soldiers, the phagocytes, to the area of the battle. So if the complement gets going around where there's an enemy, it'll break apart, and part of the little parts that's like the C3A will go out and attract some of the white blood cells to the area. All right, let's ask a question. Complement can destroy an invader by A. Manufacturing histamine. B. Manufacturing antibodies. C. Forming a membrane attack complex. D. Releasing perforin. Or E. All of the above. You got it. Of course, C. Forming a membrane attack complex. Okay, so which describes a pyrogen? A. It produces a weaker immune response. B. It signals the liver to release more zinc. C. It causes an increase in body temperature. D. It signals the kidney to secrete more iron. Or E. All of the above. Well, of course, C. It causes an increase in the body temperature and therefore will produce probably a stronger inflammatory response, a stronger immune response. Which of the following is a chemical signal specifically between white blood cells? A. Interleukin. B. MAC, membrane attack complex. C. Iron chelators. D. Exotoxins. E. Perforin. Well, really, we've only talked about a few of these, but A. Interleukin between white blood cells. What is the job of a dendritic cell? Can you think back? Okay, it's a white blood cell, so it's one of the soldiers. All right, not a granulocyte. It's kind of interesting. Does it present antigens, repairs damaged tissue, produces a membrane attack complex, manufactures antibodies, or releases perforin? So that's the one that presents antigens to the T lymphocytes, right? Yes. So repairs tissue, not so much. And membrane attack complex is only by complement. Okay, phagocytes sometimes have a hard time grabbing onto an invader. However, it is easier if the invader has already been tagged with sticky old complement. The process of attaching complement to an invader so that it is easier to be phagocytized is called what, or to make tasty? Interferon, cytokinesis, opsonization, degranulation, hematopoiesis. Well, hematopoiesis is to make blood cells. So we're talking about opsonization, right? To make tasty. Okay. And again, here we see there's the white blood cell, and sometimes they have a hard time grabbing on. And had that bacterial cell been attached with complement, it would have been easier to grab onto. Poor guy has a difficult problem there. And by the way, this would show how it wraps its membrane around the double diplococcus structure. I love that. Okay. So, little bit about macrophages. Like I said, they are the garbage cans and they are scavengers. And they're sentinels also. They go looking around, looking for foreign material. Sorry, just look above. So, macrophages, they survive weeks or months and they regenerate their lysosomes, which contain lysozyme. So, if they do eat an invader, they digest it, they break it down with lysozyme inside their little lysosomes, but they never die. A lot of the white blood cells don't survive, but these guys do. They're always present in tissue, and they're very good at calling in reinforcements. Now, they can become activated macrophages to increase their power. And so, for example, they get together and they talk to one another. And if they find that, and yeah, I know it's weird, if they find there's not enough of them to, wrap around an invader or get around something that is not self, they will fuse and form giant cells. It's rather freaky. So you get this thing going on here, these giant cells, giant cells. So these macrophage giant cells also might contain T cells, and sometimes they form a granuloma. So the body tries to wall off invaders, and the macrophages try desperately and valiantly, I can't say it, bravely. I'm stuttering like Porky the Pig here. They're very valiant at trying to take out the invader, but sometimes they can't. So they fuse. They try the best they can, and they form these granulomas. By the way, here you can see it in some lung tissue. This isn't just any granuloma caused by all the fused macrophages and some other white blood cells, but this is actually what happens with Mycobacterium tuberculosis. And they're not the only ones, but sometimes people get these little granulomas and it shows up somewhere and they're like, what is this? And that's what's going on. It's the macrophages trying so hard to wall things off. By the way, so this the Mycobacterium tuberculosis actually would be living in the macrophages, and they can spill out and become active TB. But while if the body keeps them under control, they stay in the granuloma. Okay. Neutrophil, sticky pus, and green snot. I would like to provide you with wonderful dinner topics for Thanksgiving, so here we go. All right, so neutrophil cells, these are phagocytes, as I mentioned earlier, and they're very rapid. They get out there and they fight the good fight. They don't survive, but they do play a critical role in the early stages of inflammation. So the first part of the battle, inflammation is the battle. Okay, so they are the first to be recruited from the bloodstream. So they get captured and they leak through and they get to the site of damage, to the battle scene. They are more powerful than macrophages, but again, their lifespan is very short, just a couple of days in tissue. They release all of their granules and then they don't survive that. They kill microbes by phagocytosis and again releasing all of the granules because those chemical weapons are powerful. Whenever you see a wound with pus or discharge, okay, well, the pus, the sticky, sticky pus, that is well, it's going to contain a lot of things. You're going to see something like this is going to be all kinds of white blood cells as well as the invader, the bacterial cells. But mostly all of this that I'm looking at here are dead neutrophils. And the reason, so neutrophils produce a lot of pus. They're part of the pus. Okay. And the stickiness of pus is actually DNA. So neutrophil death, the dead cells, they lyse, they break down in the battle scene, and the DNA is released, and that gives pus its sticky consistency. By the way, older neutrophils give kind of a yellow and then even later on a green color. So the older the infection, the darker the color. So this little one has had a sinus infection for a while. Poor baby. So yeah, it turns out as the neutrophils die, they go from kind of a clear to a white to more of a yellow to a greenish. I know all you wanted to know about snot. So anything that produces or generates pus is called a pyogen, not pyrogen. Pyro, think pyromaniac, fire, fever. Pyo is pus. Pyogens are bacteria that produce pus. Okay? And there are several. You have a lot of the sexually transmitted infectious disease causing organisms that do. But here's one, Streptococcus pyogenes generates pus. Or sometimes we refer to this as group A strep, causes strep throat, right? All of that. That's where it gets its name. All right. Let's put it all together. And now we've talked about the players and the places and the chemicals. Now let's put it together and really talk about the second line of defense. And ouch. Okay, so all of this is coming together. All of it. Everything that I've talked about comes together in the second defense, line of defense in inflammation. Okay, inflammation is your symptom maker. If you have a pathogen, an infectious disease, and you have symptoms, look to inflammation. Okay, something's going on there. Okay, so the inflammatory response is really the second line. So the purpose is to contain the site of damage, localize what's going on so dangerous elements don't escape. So you have to kind of contain the invader, eliminate the invader, and restore tissue function. So repair, that's what's going to have to happen. But whenever you look at inflammation, I want you to think battle zone. Okay, and wherever there's a battle zone, there is collateral damage. There's a lot of tissue, not just the white blood cell soldiers. Oh no, when they're releasing their granules with all those chemicals, lots of damage occurs. Now, the bacteria and viruses will also cause damage, but the battle, okay, and it's not only the enemy, it's the battle. So, symptoms of inflammation, swelling, redness, heat, pain. Okay, you're probably familiar with this. But sometimes it can be so severe that you can lose function, even cause paralysis. Okay, so let's put it all together. Let's see what's happening in the battle zone, in the war zone, okay? So the inflammatory response involves a cascade of events. So first of all, let's just say you have injury. So your tissue has been breached. Now, this could be because the bacteria or the viruses are damaging your tissue directly. It could be because of a wound, something like that, but there is damage here. The cells, okay, the civilians here, your cells, they're going to send out SOS cytokine signals to draw in the white blood cells, and here they are. And you can see them traveling through, and like I mentioned before, the white blood cells are going to get sticky. They're going to attach to the endothelial cells and come on through and find the invader. So the migration of the leukocytes from the bloodstream to the tissue, to the war zone. Now, granulocytes, many of the granulocytes, in fact, most all of them release that chemical known as histamine. Histamine is going to dilate the blood vessels. So they're vasodilators. Why is that important? Well, that opens up the interstate, the road. If this swells and gets bigger, then more cells, more white blood cells, and even the red blood cells get to the area. Now, the red blood cells shouldn't leak through, but they can carry their oxygen. So that sends in more troops and support. Okay. It also will help to make the endothelial cells a little more leaky. Okay. So this is going to increase the inflammatory response, the histamine, which is why when we have too much of an inflammatory response, we take antihistamines, right? So this is going to mean greater blood flow. With that, heat, redness, swelling. Okay. Leakage of fluid, redness, swelling, and pain. If you have nerve endings here, there's going to be pressure. That's going to give you pain. Now, you're also going to get clotting factors that are going to try to wall everything off and try to keep you from bleeding out, okay, and fix the problem. So I just wanted to show you battle zone, inflammation, redness, to everybody rushing to the area, the inflamed area. You're going to have a lot of dead neutrophils and a lot of other tissue debris. Some bacteria are so good at producing all of this that, again, you do have pus. So some bacteria, so much so that you get pus. Okay? And then there's repair. You got to go back and repair these areas. Sometimes the result of a huge battle is scar tissue. Scar tissue does not replace the original tissue. It is different. It is hard and it is just fibrous. Okay? Okay, what are the consequences? Well, and we'll talk about that in just a moment. Now, what if the damage or the inflammation is going on in a mucosal area? So mucosal inflammation. Then you get a little something extra. So you still get the swelling, but it may not be noticeable, but it'll probably be felt. You're still getting the swelling and probably the pain. You may or may not see the redness. So with the influx of fluids also is going to be part of the mucosal inflammatory response. With the fluid, you get more mucus. Now here you have, now this is a mucous membrane and you have goblet cells and they're going to be producing extra mucus here. So you're going to get so much mucus and hopefully you have a way to get rid of that. So yes, in the respiratory tract, you're going to get discharge, pain, and coughing. How about in the GI tract? Same thing. You get discharge, and you get pain, but you can get cramping and diarrhea as a result. So I always thought that was hilarious. Probably not when that's you, but okay. Okay, how about some laughter? And if you need to take a coffee break, this is a good time. Laughter is the best medicine, unless you have diarrhea. Isn't that the truth? Anyway, so if you need to take a coffee break or something else, okay. You back? Good. All right. So let's go back to the inflammatory response. As I said, this is a battle zone. There's a lot of innocent cells that are going to be destroyed. And if it's serious damage, you're going to get scarring, permanent damage. Okay. This just shows the scarring that can happen if you have, say, a sebaceous gland that's infected, acne, anything like that. And it can get pretty severe. You get the inflammatory response going on, and yeah, you get permanent scarring. Scarring can also block the reproductive organs and cause sterility and something called an ectopic pregnancy. So there's a lot of the reason that my boss went into research, by the way, was because, and I said he studied gonorrhea. Why? He was interested in fertility problems in women. Gonorrhea, chlamydia are big culprits of causing sterility in young women, and it causes trouble in men too. But this is one of the biggest issues. Okay, so let's say you have intercourse, somebody, your partner has Neisseria gonorrhea will hitch a ride on sperm cells. And you can ride all the way up to the fallopian tube. Okay. Now, normally, of course, eggs are released once a month and they come down the fallopian tube to embed in the uterus. But now we have gonorrhea up here, Neisseria gonorrhea, and a big inflammatory response. So you're going to get damage and you're going to get scarring. So that scarring is going to block any of the eggs from getting through, sperm from getting through, and sterility. Okay, permanent scar tissue. Remember, the scar tissue is not original, it is tough and hard. Okay, doesn't allow the passage of materials. So again, eggs don't get through, sperm doesn't get through. Sometimes what happens though is that the egg gets to a certain point and it can be fertilized, but it can't go any further. And then you can develop an ectopic pregnancy where is basically the embryo implants someplace outside of the uterus. Okay, sad day. So what should you do to prevent this? I know, don't be afraid to talk about sexually transmitted infectious diseases. Chlamydia and gonorrhea tested together, where there's one, there's probably both. And when you start to see a discharge, okay, if or not you, but also your patients and your family, especially young girls, everybody pretends that they've never been exposed to an sexually transmitted disease, please. Okay, unless they're living as a monk. Okay, there you go. But chances are everybody has. And if you're unfortunate enough, it might be one of these organisms that can cause sterility. So at the first sign of symptoms, you need to take an antibiotic. The more the battle occurs, the more it goes on, the more inflammation, the more damage, and the more infertility you're probably going to be, you're going to be at risk for more infertility. Okay. A greater risk for infertility. Sorry. So please, 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 there are certain things in microbiology that, okay, I want you to pass on to people. This is one of those things. You need to be diagnosed and treated with an antibiotic before the damage gets too much until it gets, you know, certain parts become irreparable. And this is something there's nothing you're going to be able to do about that. Okay. Um, Hopefully the other fallopian tube is still kicking. Okay. So sterility is a big thing. Same thing with hepatitis. You get viruses that infect the liver cells, okay, causes an inflammation or hepatitis. Hepatitis A, B, and C all will cause this inflammation. This is actually a picture of a patient who had hepatitis C. So this is why we have vaccines to prevent it, but also we want to treat it. You can see all the liver fibrosis. Blood and doesn't move through this organ hardly at all because the fibrous material won't let it pass. So you get backup and you get, you don't get all of the nutrients, all of the functions of the liver, and you get varicose veins even. Okay. How about in the mouth? Inflammation. Yes. So here we have a normal healthy gum, but we have bacteria here that are causing an inflammatory disease in your gum line, and you get red, swelling, and heat, and maybe pain, and it pulls away. Unfortunately, there are even more bacteria now that can move into the deeper crevices, the ones that cause the really smelly, they're anaerobic bacteria, and they will be even smellier than the ones that we're infecting up here. And the inflammation can get so severe, the battle zone can get so deadly that you can have bone loss. So here you can see this is really a terrible case of periodontitis. So you have swelling of the gums, but again, it's pulling away, and this person needs a lot of help, right? Right. Okay. Again, when trying to find why your pathogen causes a certain symptom, really probably most all of the symptoms, most if not all of the symptoms can either directly or indirectly be tied into inflammation. So insert your disease here. So it's why we want to treat our infectious diseases very quickly so that you don't get permanent damage. Okay, some questions. This is not part of the inflammatory response. Promoting healing, cleaning up damaged cells, releasing histamine, causing vessels to dilate, secreting mucus and mucosal membranes, blocking all symptoms. Yeah, E. Blocking symptoms is not what the inflammatory response does. When there's a battle, you have symptoms. And think inflammation, battle. Battle zone, war zone, all the troops are coming in, swelling, redness, pain, inflammation. Okay. The process of let's ask let's go back and ask some more questions. Hey, the process of producing red blood cells, or excuse me, blood cells in general. Epitopes, apoptosis, inflammation, hematopoiesis, cytokinesis. Hematopoiesis is correct. D, good for you. A cytokine that produces a fever. Pyrogen, pyogen, interferon, hemolysins, lysozyme. Right, fever, heat, fire, think pyrogen. Okay, third line of defense, the adaptive immune response. Okay, if you need to go get coffee, now's the time. There's a lot going on. Okay, so, all rats, they're not working. Oh, this was a wonderful video showing white blood cells in a lymph node. The little green ones, they were bumping into and trying to find um, trying to find the right dendritic cells and B cells, and they were all interacting with one another in this particular video. Ah, shucky darn. Okay, well. As we get into the third line, I really need to start with talking about the natural killer cell because this one blurs the second and third lines of defense. So here we have a building being blown up. So let's say this was self. This was, something that was part of your city, but now it harbors the enemy. It's been taken over. Well, natural killer cells blow it up. Okay, they kill the cell. They kill the cell that's infected or non-self. It has enemies or it doesn't belong anymore. So, the natural killer cell is a lymphocyte, and you're going to find that it's an awful lot like a particular type of T lymphocyte called a cytotoxic T lymphocyte. But it's not as specific and picky. It just has to find something that's non-self and blow it up. So, it's a non-self demolition squad. Okay, how do they blow up a whole cell that doesn't look like self? Well, first of all, what kinds of cells do they destroy? What would be non-self? Well, if a cell has been invaded by viruses or bacteria, okay, then that's got to be blown up. Cancer cells, they have become traitors. Okay, so they need to go. Transplant cells from organs, unfortunately, they look like non-self, and a natural killer cell will go after it. How do they do this? They are carrying chemical weapons, even though they're not considered granulocytes. One of their chemical weapons is called perforin. It's a protein, and it literally punches or perforates the membrane of the infected cell. So this would be one of your cells. Let's say this was a cell in your nose that had cold viruses inside. Here you have the natural killer cell. You can't see the chemicals, but you can sure see the big holes. Okay, so this is harboring viruses. It must, we must punch holes in it. That's not the only chemical. This one punches a hole, but there's also another chemical in case this one doesn't work. When I was learning immunology, we didn't know about granzymes, but granzymes, again, tell the cell to kill itself or induce apoptosis. Apoptosis is cell suicide. So you have been infected, you have been taken over by viruses, you must die. Okay? By the way, this could be a cancer cell or a transplant cell. All right. Natural killer cells, by the way, their ID tags are different than the ID tags that we're going to talk about later on the T cells. The T cells are CD4. CD1234 cells. Remember we said, oh yeah, HIV loves CD4 cells. That's how the HIV viruses grabs on to our cells, and they grab onto a white blood cell called a CD4 cell. I remember. Yeah, well, okay. So HIV will go after them, but they can't attack the natural killer cell because natural killer cells have a different kind of receptor known as a CD8. This is why we think Magic Johnson survived HIV and didn't really, he never died from AIDS, he recovered. We're thinking that he just had amazing CD8 cells, natural killer cells, that took care of the problem for him. I know, huh? Ah, good. This video's working. Yay. Okay. So this is a cancer cell in blue. What's the red cell? That is the natural killer cell. It looks like it's on fire. Remember what I said? When white blood cells get really excited and they're going to phagocytize or attack, they get fluffy like that. And look at that go. So no wonder natural killer cells. Again, not only do they go after cancer cells, but if there's a virus in there, or something like that. It's too bad they can't always do the job we would like them to do, but they try. If you treat them well, they will. Okay. Okay, let's go full blast to the third line, which is the specific adaptive defense of the host. This line has cells that learn, they get better, their offspring are smarter, they inherit even better genes, and they're even better and faster at taking out the enemy. But they're very picky, very specific. So, all right. All right. So they search, specifically identify, destroy, and remember. Okay. For lifelong immunity, hopefully. All right. Adaptive, to learn. Okay. And this develops throughout life. Again, the response is quite specific. Lymphocytes that we're going to talk about here in the adaptive immune response only respond to specific non-self ID tags, so the little epitopes. Okay. And they're really, really picky. So if you're a lymphocyte and you're in the lymph node and you only identify an ID tag on a chickenpox virus, well, you're going to ignore the cold viruses and all the other herpes viruses. You're very specific. Okay. So again, the lymphocytes, being very specific, only talk to other white blood cells, also trained to work on this specific non-self invader. So, okay, you're back in the lymph node, and you're only going to talk to other white blood cells that work on chickenpox. You're not going to talk to the ones who work on, like mono or cold viruses. No. Okay. All right. So, and again, it produces memory. It learns, it gets better, it gets stronger, it gets faster each time it is exposed or challenged. So every time it is exposed to the chickenpox virus, it gets better. Okay? The first response to the antigen is the primary response, and I'm going to talk about this a little bit later on. It takes about 7 to 14 days for the first response and to get all of the white blood cells working to fight the infection takes a little while, but it does remember so that, if encountered again, the stronger secondary response is really fast, okay? But this only happens if you have memory from the first response, memory cells. So if you do, the memory cells launch into action, hopefully before any symptoms. Yay. And this can happen within 3 days. Yeah, probably don't even get a symptom. Vaccines will give you memory, hopefully without disease. More on that later. So there are 2 basic strategies for countering foreign materials. So what I'm going to talk about, okay, so these strategies are kind of like 2 branches of the military. So way, way back when we sort of had the Army and the Navy. Okay. So that's sort of like this. We have 2 branches of the military to fight this very specific battle. And again, you're going to find all of the soldiers in the lymph node, but some will be Army, some will be Navy, or some are part of the humoral response, some are part of the cell-mediated response. Okay. The soldiers in the humoral response, or humoral immunity, are the B cells. B's are humoral. Okay? B cells are B lymphocytes. Now, B cells are born in the bone marrow and they are educated in the bone marrow. B for bone marrow. That's not exactly accurate where the bee comes from, but you don't want to know about the chickens they did research on. Okay, so the B cells specifically have attached antibody receptors, and those receptors identify specific invaders. Okay? So, and their job is to go after extracellular antigens, extracellular invaders. So, if anything's floating around, maybe eventually it'll get into a cell, but it hasn't yet. So, bacteria floating around, toxins floating around, viruses in the bloodstream, in the tissue fluids, maybe they would like to infect and get into a cell, but they haven't yet. They're all extracellular. Okay, so what happens is the B cell here will identify this very specific invader. Notice it has lots of different epitopes, greens and pinks and variations of each, but it has specific receptors and it matches up to the yellow part. And there you go. Now, if you're wondering if these look like clothespins, they are. I have a laboratory where I use clothespins. So it works, guys. But again, like I said, I have to make my own animation because I didn't have you buy a textbook. That would have been easier, but nah, who likes easy? Okay, the other military branch, the cell-mediated immunity. Those soldiers are the T cells or T lymphocytes. They are also born in the bone marrow. Of course, remember, all the cells are born in the bone marrow. But these guys are educated in the thymus. T for thymus. They also have specific T cell receptors, sometimes known as TCRs. Their target, basically, these are the guys going for intracellular pathogens. Okay, anything that got past the bees, and okay, now they're in the cell, or maybe the cell has turned bad. It's become altered or become non-self. So, invaded viruses that have invaded, bacteria that have invaded, a cell that has been, that has gone wrong for whatever reason or another is cancerous or non-self. Okay? And there's the T cell. Notice both of these, let's say this is chicken pox, and this is all in the lymph node. The B cell, there's a B cell here that would recognize the chickenpox viruses while it's kind of free floating. And then the T lymphocyte would also recognize this particular virus as well if it was invading a cell. So if it was inside a cell. Okay. So it's all about taking out the antigen. Okay. And again, some invaders, some antigens have multiple epitopes. So B cell lines, the B cells have specific B cell receptors. The T cell line has specific T cell receptors. Okay. And again, these B cells and T cells would be the only ones in the lymph nodes working on the chickenpox virus. They would ignore everything else. The other T and B cells would be working on the cold virus and the adenovirus and the monovirus. Okay. And these guys would ignore this guy. Okay. Different receptors. Okay. Different color. I'm color coding it just to make it, you know, tangible. Okay. So, all right. Now, if you are a white blood cell, if you're part of the military, you better have an ID tag of your own. You better show that you are self. Okay? That is actually the major histocompatibility complex. Maybe you already are familiar with this. When people get a new organ, they want to match ID tags or the major histocompatibility complex. There's a little more to it than that. We don't have a whole lot of time to talk about it. Just know that there are two types of MHC molecules. The first class, and again, you don't have to know this, is FYI only. The first type of ID card, all self cells carry it. If you are, look at all the cells in your body, if they have a nucleus, they have that ID card. And okay, if they are military and they are antigen presenting cells, then they have another type of ID tag. Okay, another major histocompatibility card. That's all. So this would be the major histocompatibility complex Class 2 would be carried by a dendritic cell or a B cell or a macrophage. Okay, because they all, I didn't mention this yet, but in addition to dendritic cells, some B cells and macrophages are antigen presenting as well. So again, not only are they ID tags, but this is a way to present an antigen to T cells as well. I know this is kind of busy. I made this cartoon a long time ago, but here you can see, here's the dendritic cell. He's carrying his own identification. But this other card, because he's an antigen presenting cell, he's presenting the antigen that he chomped up. So here's the antigen. He broke it apart, and then he's going to present it to this T lymphocyte, okay, called the boss. Okay, so let's talk about the T cells for just a moment. When you have this interaction, the dendritic cell finds the invader, processes it, and submits it through antigen presenting. Okay. The T lymphocyte with the right receptor kind of docks up with it. Okay. And we're going to have a war, basically. Okay. Against everything that looks like this. So the cell-mediated response, as I said, protects against intracellular threats, viruses and cancer cells, okay? Or intracellular bacteria. If a cell has a lot of intracellular bacteria, sometimes cell-mediated immune responses will go and blow up that cell too. So it detects non-self or altered cells. The original cell, this is actually what's called you get kind of a naive T cell, and he's a CD4, and he's the one that will interact with the dendritic cell, the antigen presenting cell, and it becomes activated. Okay? And for this to happen, you have to have the handshake, the antigen presentation to the receptor, that's the handshake, and a conversation. Okay, got to have the conversation. Cytokine, cytokine. All right. And then you have war. So here's another picture. Here's a dendritic cell, antigen presenting to a T cell, and it tells the T cell, and the T cell releases cytokines, tells the dendritic cell, hey, I am trained to take care of this problem, and the dendritic cell says, well, then take care of the problem, let's have a war. And the T cell then will only go after this pathogen with this epitope, so just chickenpox, okay? So this causes a proliferation of the T cells. So what does that mean? This T cell that's been activated will go off and replicate and form lots and lots and lots of kids. Okay? All of which are trained genetically and have the receptors for chickenpox. So, okay, so some of the kids are going to be different kinds of cells we call effector cells. Okay, so some of the kids are going to be helper T cells. So helper T cells are CD4. Their job is to help. They orchestrate. And not just all of the T cells, but also they will help the B cells as well. So they orchestrate all branches of adaptive immunity to take out this one type of invader with just this one epitope. So the chickenpox with the chickenpox ID tag. And again, the helper T cell will produce even more T cells. It will activate more phagocytes. It'll activate macrophages and the B cells too. I know we're not even in the humoral response, but it will. The T cells will boss around the B cells if they can, and that's a good thing. One of their deadliest kids, okay, one of the effector cells that is produced from this interaction is a child, offspring by the name of the cytotoxic T cell. Okay, and again, this is like natural killer cells. It's a CD8. It's a lot like the natural killer cell, in fact. All right. So here we have, okay, down here we have the here's the infected cell. Here we have a cytotoxic T cell, and again, this infected cell knows it has chickenpox, and it's presenting chickenpox ID tags. The offspring from the earlier helper T cell, the cytotoxic T cell, docks up, shakes hands, realizes this is an infected cell. It must die. Okay, so how does it die? Well, Okay, so it destroys altered cells by releasing two deadly substances. And what would that be? Perforin? This really shows it perforating, and granzymes. And this shows again another picture. So this acts exactly like a natural killer cell. So Miss Eden, what's the difference between a cytotoxic T cell and a natural killer cell? Okay, because they're both CD8s. They both, blow up altered non-self cells, cells that have been infected, taken over, what have you. True, they both have the same kinds of chemical weapons, perforins and granzymes. The difference is cytotoxic T cells are only made to a specific threat. Okay, so it has to go through all of these processes. It has to answer to a helper T cell and this activation when there's this handshake and conversation. It's really important. Now, if you're thinking, why go through the trouble? Why don't you just have cells that go around blowing up anything that looks like non-self? Well, then you can have some very nasty autoimmune diseases and die from that. So you want to make sure you have checks and balances. Everybody checking, double checking, handshakes, conversations. Okay, we're only going to make a lot of these cells that specifically attack chicken pox infected cells and nothing else. Okay. By the way, if you get a transplant, the transplant organ will probably have, again, a cytotoxic T cell coming after it. It's a weird process and where it's not fully understood, but one of the things we can do when we want to stop these CD8 cells from releasing granzymes and perforin is we give cyclosporins. So just as a little side note. So that's one of the therapies with your transplant patients. There's also a delayed hypersensitivity T cell. I don't have time to talk about it, but this is what causes a delayed reaction when you have a TB shot, which isn't a vaccine. It's just you've just got little particles of tuberculosis bacteria. And and if you get a big swelling, that means you're reacting in a big way. Okay, and then there's also the suppressor T cells, and they do the opposite of the helper T cell. They go through and block everybody. So the helper T cell says, let's start a war, and the suppressor T cell says, let's have peace. Okay, because and this happens when everything is done, the war is over. Okay, now the other cell that has to be made in addition to the effector cells that are involved in the battle, memory cells, memory T cells. Very important. So if it ever comes back, you're faster, you're stronger. Okay, so here, this one's working a little bit better. So here's an animation. You have the T cells, and you can see the T cell is having handshakes. It's actually trying to find the right dendritic cell, and if they have the same receptor, okay, they will shake hands, and then the T cell then will also interact with the red B cells, which we're going to talk about in the next form um, of specific adaptive immunity. So all of this is in a lymph node of a mouse. And look how excited the T cells and the B cells are. And they're all coming up to the dendritic cells to find out if they're going to have the right receptor, if they should get involved. This isn't chickenpox, but let's say that the dendritic cell has a chickenpox virus epitope, and it's presenting it to B and T cells. So they're bumping into each other, trying to find the right lymphocytes to fight in the war and then start a battle. Excuse me. Okay, let's talk about the humoral response. Okay, it doesn't have anything to do with humor, but why not? So the humoral response, like I said, protects against extracellular threats or viruses that haven't invaded yet. So if they're floating around, these are the guys to go get them. So here we have, in fact, here we have the little viruses floating around. So what is the B cell and the B cell line going to do? Oh, let me back up just a little bit. So this B cell will, when it becomes activated, it will make a lot of plasma cells. Plasma cells are antibody factories, and they will churn out tons and tons and tons of antibodies, which will attach to and label the enemy for destruction. Sorry. So let's kind of go through here. Sorry. Okay. So, humoral response. What does it do? So remember, the humoral response, it has the soldiers that are the B cells. But if the T cells talk to the B cells, the helper T cell, if it gets involved, then the B cells will make memory. If the helper T cell does not get involved, then it's kind of like the whole military is unsure to declare war later on. It must have that helper T cell to say, yeah, this was a big problem, a big threat. We need memory because it'll probably come back. So without the helper T cell getting involved, without that portion of cell-mediated immunity, no memory. Okay? All right. So the B cells, when they become activated, and sometimes and what they will do is they will phagocytize the same invader. And again, if they talk to a T cell with the same receptor, and they both know, okay, we're both trained to take out chicken pox. Let's do it. And this will charge up the B cell, and they will divide and proliferate into plasma cells. And again, these are the little antibody factory cells. So they make lots and lots and lots of antibodies. Now, the antibodies do not destroy directly. They don't hurt anything directly. They tag non-self things for destruction, label them for destruction. Who destroys them? Phagocytes will come along as well as granulocytes. So, yeah, that's what the antibodies are supposed to do. Okay. Also, memory B cells are made, but only if the helper T cell was involved. Again, you need both branches of the military to be involved for a full-out war. Okay? So without the helper T cell, the B cell will have a battle, but not a war. Not a long-term war, and it won't remember. So it can come back and cause disease again. So again, the B cell may act alone if necessary, but without the helper T cell, no memory. Have I said that enough? Now, when does the B cell have to act alone? Normally, this happens when the antigen is not a protein. Not a protein, shouldn't be proteins. So, for example, if the invader has a capsule, remember the sugar capsules? Yep. The T cell cannot respond to sugar, right? And bacterial LPS, T cells can't respond to that either. So in this case, you get very poor memory and a poor immune response. If there's a protein to be detected, if that is the ID tag, the immune response is good. By the way, all of this is going to be relative when we talk about vaccines. Okay? All right. So let's talk about the antibodies. A fancy way of saying antibodies is immunoglobulin. That's when we want to sound very fancy and smart and educated. So what can immunoglobulins or antibodies do? Well, with the help of complement, these little kind of square things, the antibodies will also attach to complement, and they then will be very, very, very sticky. So when you have a phagocyte, the phagocyte very easily brings in the invader. Okay, so opsonization is even better if you have an antibody there. So, okay, what opsonizes? Complement, antibodies, better yet, complement and antibodies. Why do you want to opsonize? Opsonize to make tasty. It makes it easier for phagocytes to grab onto and eat the invader. Okay, also agglutination. Bacterial cells and viral cells and toxins, if they get all clumped up, they're a big sitting target. Okay. And gosh, let's say they're intracellular parasites, bacteria, toxins, or viruses. And these would be bacterial toxins. If there are antibodies, then they get all gummed up, they're all clogged. And how are they going to attach to the host cell? How are they going to attach to the host cell receptor with all these antibodies stuck on them? Yeah, they can't. So neutralization. Yay. And of course, the antibodies do help complement. So it helps complement. I like this. So little cartoon. Bacteria trying to avoid the immune response. So here we have, which cell is this? It's a white blood cell. Yeah, which one? Lymphocyte. Good. B or T. Not exactly a B because he's making the antibody. So this is a plasma cell that came from a B lymphocyte, probably the guy in the back. So, okay. So I'm hit. Go on without me. But again, the antibodies don't destroy. He's labeled for destruction though. Okay, so here are my clothespins. And this is just the nature of antibodies. I know, bear with me. Okay, the structure and properties are critical for the function of the antibodies. As I said, sometimes antibodies are called immunoglobulins, or a capital I, little g, and then there's usually another letter to tell you what kind of antibody or immunoglobulin class we are referring to. So these are Y-shaped proteins and they have two general parts. I'm not going to talk about the heavy chains and the light chains. If you've ever learned that, it's not going to come up today. But there's the variable region that is very specific. This is the yellow part. So this would only attach to yellow epitopes. It would not touch and attach to the purple or orange or pink, right? And they have two identical arms that will bind the antigen. And then there's a stem area here, and this is constant, and this one a lot of times is sticky, and our own cells, like phagocytes, will attach to the sticky part. So let me back this up so you can see that here. Okay, so yes, for opsonization. Yay. All right. Moving along. 5 types of immunoglobulin classes, or Igs. There is IgM. This is the biggest, the monster, and I'm sorry it comes up last in this illustration, but it is huge. It's a pentamer. What does that mean? Pent, like penthouse, 1, 2, 3, 4, 5, 5 sides. So you have like all of these little Y-shaped antibodies all stuck together. So, wow, look at all those sticky little arms sticking out. So this is going to be a great opsonizer. It's going to grab onto a lot of things. The IgMs are usually the first in. When we look into plasma and we want to see if you have been exposed recently to hepatitis because of a needle stick, if it was recent, we'll see a lot of IgMs. Okay, we'll see a lot of those in your blood, in your serum. If it's a little bit later on, like it's like, no, you've had this for a while, then we'd see another type, okay, called the IgGs. So, and the IgMs do work as receptors on the B cells. You don't need to know that. So again, the IgG, if it's been a little bit later, like maybe it's your second, third, fourth exposure, what have you, been a while, then you're going to have a lot of the IgGs floating around. So yeah, most of you have been exposed to chickenpox. So you have anti-chickenpox antibodies floating around, and in your serum, you're going to have IgGs. So when they're checking your serum to see if you are that you have a immunity, they look for IgGs against chickenpox, although I don't think they do that really. So IgGs are the major type in the blood. This is really important for mommies because the IgGs cross the placenta. The others, not so much, okay? So while the baby is still in the womb, this is the IgG. It doesn't last for a long time, but it's not bad. It actually sticks around for 6 months. So after the baby's born, how long will that baby have mom's IgG antibodies to protect him or her? 6 months. You got it. Good job. Okay, IgA. IgA is just, usually it's either a monomer the meaning it'll look kind of like this one, or sometimes it comes up as a dimer. If it looks like this, you'd find it in the blood. If it looks like this, it's in mucous membranes, or tears, or saliva, or hey, the baby's been born and it's got IgG antibodies circulating around the bloodstream. But what about in the gut? Baby's taking in milk and other things from its environment. Is there anything to protect baby's mucosal surfaces? Well, yeah, IgA from mom's colostrum and milk. Okay, so the IgGs will hang around for 6 months, kind of circulating everywhere. But in the gut, you have IgAs. Breastfeeding good. Okay, if you can. Okay, IgEs. IgE monomer associated with parasites and allergies. Usually IgEs will be very attached to eosinophils, those granulocytes that are seen when there are parasites or allergies. So IgE antibodies and eosinophils, parasites and allergies. Okay, link the two. IgD, I'm not going to talk about it, but they are receptors on B cells. Okay, so, okay. So we already talked about protective outcomes of antibody-antigen binding. We have the opsonization, which enhances phagocytosis. We have the aggregation or agglutination, where the two arms bind up different antigens. We have neutralization that stops deadly bacterial toxins from attaching or even binding to the host cell, to the target cell. And here we have another way. So let's say the bacterial cell was about ready to attach and it can't. So there's our neutralization. Woohoo. Okay, it can't attach to your cell. What could this be? Oh, this could be like Yersinia pestis that causes the Black Plague. So I know I'm dramatic. Okay. And then, of course, there's the complement activation, uh, uh, complement system activation, and something called antibody-dependent cellular cytotoxicity. What's it? Okay, let me just show you here. So this is the complement system activation, or excuse me, the antibody-dependent cellular cytotoxicity. This means that you're going to have to not only have an antibody there, but you're going to have a big natural killer cell or a granulocyte packing chemical weapons. Maybe the invader is too big, okay? And like the phagocytes can't eat it, it's too big. So what do you do? Well, if you could release and kind of release some of those granule chemicals, then maybe we can do this. So here's a good example. Here's a parasitic worm, a flatworm, a fluke, known as a schistosome. Actually, it's the larva stage, a blood fluke. So you don't need to know the name of it, but it's a parasite. It's huge. Here's the outline. What is this all around it? Oh, glad you asked. Well, okay, so these little cells are actually eosinophils. Now, imagine, if you will, IgE antibodies have tagged it for destruction. They can't hurt it themselves. Antibodies don't destroy, they tag for destruction. But then a granulocyte or a natural killer cell attaches to the constant region. This activates it and it releases its chemical weapons. Not impressed? Okay. Here's a living parasitic worm. And it's probably, you can't see the antibodies, but you can see the granulocytes and the eosinophils coming in for destruction. Isn't this cool? Eosinophil IgEs versus worms. How long does it take to make enough antibodies so that you are protected? All right, well, again, you have your primary response and you have your secondary response. Over here, we have the antigen, and again, it doesn't find every B cell. The B cell that is involved has the right receptor. It is activated. You get B cells that make a whole bunch of memory B cells and B cells that become plasma cells. The plasma cells will make antibodies, tons and tons of them. The first exposure. The memory cells will wait, and if it ever comes back, then one of the memory cells replicates very quickly, and you will make more memory cells, but even more plasma cells with even more antibodies. A fast, fast, fast response. And how long does it take? Again, about 7 to 14 days for large amounts of antibodies to be made. By the way, is the 7 to 14 days how long we take antibiotics? Yeah. So for a lot of the antibiotics, they're just there to keep the bacteria at bay until a good, healthy immune system makes enough protective antibodies, primary response, okay, to take them out. Okay. All right. And then also, if we get a vaccine, we say it takes about 7 to 14 days before you're actually protected from like real influenza. Now, during this time, the person may be sick, possibly seriously so. Although the immune system is actively responding with an inflammatory response. But remember, inflammation equals symptoms. So you're probably going to be feeling sick until you stop. Now, some activated B cells do continue dividing, but again, others will differentiate to form the antibody secreting plasma cells. Okay. Now, some of the B cells, then, when they divide, they become memory cells, and that means you remember. So the memory cells will respond very quickly. So if it ever comes back, if the invader ever comes back, the memory cell says, oh, yeah, I know what this is. So it's more exposure. You're going to get a stronger and faster, mostly IgG response. Like I said, it can be so fast and so strong, you're maybe asymptomatic. And the enemy gets removed before you get a big inflammatory response, which is awesome. So just because you've never gotten the symptoms of chickenpox twice doesn't mean that you haven't fought that little virus off many times. Anytime a child with chickenpox ever sneezed on you, oh yeah, you were probably fighting the battle, but you fought it quickly. So you never had the pustules, you never had chickenpox. That's why. And again, this just shows the initial response, mostly IgMs. And again, to get memory, the T cell has to be involved. But if the T cell is involved, you make memory. If it comes back, the response is so much faster. The symptoms are subclinical. You probably don't even get it. And again, the more times you are exposed, the faster the response. Okay. So, just as a reminder, just to let you know, everything in the third line of defense, the adaptive immune response, must be recognized with receptors. And you've got to have an antigen presenting cell there, okay? There's going to be a handshake and there's going to be communication with chemical signals or cytokines. Remember, there's two branches of the military. There's the cell-mediated immune response. Those are the T cell soldiers, the T cells. Okay. There are some that coordinate, some that destroy, some that halt the attack, and more so, there are those that are memory and they remember. So if it comes back, oh yeah, they act very quickly and divide even faster. The second immune response, the second military branch, is the humoral response. These are the B cell guys. And the B cells divide, they make plasma cells. The plasma cells make the antibodies. Antibodies do not destroy, they tag for destruction, and they do things like neutralize the enemy until something like a phagocyte comes and num 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 eats them up. And they remember. Okay, and this video didn't work. I'm sorry. So let's do a quiz or take a break. Take some coffee, get some coffee. Okay, break. Okay, you're back. Let's do some quiz questions. What does specific immune response mean? So what does Miss Eden mean when she says that? A, there are specific receptors that recognize specific epitope antigens. B, it specifically attacks invading bacteria or viruses. C, T and B cells specifically switch receptors in order to recognize invaders. D, the body puts up barriers to specifically block invaders. E, the body makes lysozyme that specifically attacks bacteria and viruses. Of course, A, there are specific receptors that recognize specific epitope antigens. Good. Didn't want this A here. Let's get rid of that. Oh, okay, that's prettier. This immunoglobulin antibody is found in the greatest numbers during your secondary exposure to an invader and the triggering of memory B cells. So basically, this is what you're going to find in the blood after you've been exposed the second time around. IgA, IgM, IgG, IgE, IgD. It's the IgG. Remember, those are the ones in greatest numbers, especially in the blood. These are the ones that are going to come from memory cells for the most part. Okay? In greatest numbers, IgG, C. Which immunoglobulin is found most commonly during a parasitic and allergic reaction? IgA, IgG, IgM, IgE, IgD. IgE, right, with the help of eosinophils, maybe, and other granulocytes. What is a macrophage characteristic? Ingest dead bacterial cells and debris, release histamine, produce a membrane attack complex, release perforin and granzymes, produce antibodies. Right, macrophages are the garbage cans, so they ingest dead bacterial cells and debris. Yeah, histamine would be a granulocytes. They're not a granulocytes. Membrane attack complex, that's complement. Perforin granzymes, okay, those would be CD8 cells, like natural killer cells, and its cousin, cytotoxic T cells, and produces antibodies. That would be plasma cells. T lymphocytes are educated in the spleen, thymus, thyroid, lymph node, or bone marrow. Ah, not the thyroid, but the thymus. And especially, this is going to happen in the young years. The thymus is reduced in later years, of course. It's born in the bone marrow, but it's educated and gets its receptors in the thymus. That's where it gets its receptors. So T for thymus. Okay, now the B cells are educated in the bone marrow. What does an antihistamine drug do? Okay, so you got to think, what does histamine do? What does antihistamine do? Stops granulocytes from degranulating, causes blood vessels to dilate, causes blood vessels to become leaky, boosts the inflammatory response, or all of the above. Of course, it's A. If it's antihistamine, it will stop granulocytes from degranulating, which means that it will stop the blood vessels from dilating and stop the blood vessels from becoming leaky and reduce the inflammatory response. Okay? All right. Which describes a pyogen? A, it is secreted by antibodies. B, it signals the liver to release more zinc. C, it causes an increase in body temperature. D, it signals the kidney to secrete more iron. E, it is a bacterial organism that generates pus. Right, it is a bacterial organism that generates pus. That best describes it. Okay, this immune response is necessary to destroy altered non-self cells, but doesn't produce antibodies. Okay, that is called agglutination, immunity, humoral immunity, cell-mediated immunity, political immunity. Haha, I know. Yes, so altered non-self. Okay, it's got to be cell-mediated, right? C for cell-mediated. Which of the following cells has a major role in both humoral and cellular immunity? Bosses the others around. Helper T cells, cytotoxic T cells, plasma cells, B cells, all of the above. That would be the helper T cells, CD4s. Yep, they are the ones, and without them around, you don't get memory, and you don't get a strong immune response from both branches of the immune system. Okay? Which of the following is a chemical signal specifically between white blood cells? Interleukin, membrane attack complex, iron chelators, exotoxins, perforin. Interleukin, right, A. Good. What does not happen in the lymph node to bring about the cell-mediated immune response to an antigen or epitope? So everything else is happening, but this is not happening in the lymph node. So A, the nearest CD4 T helper cell will phagocytize it. B, the antigen must be presented. C, there has to be specific cytokine signals. D, helper T cells proliferate. E, cytotoxic T cells with the perfect receptor kill the infected cells. So what is not going to happen in the lymph node? Okay, yeah, the nearest CD4 helper T cell is not going to phagocytize it. The T lymphocytes don't phagocytize, and they are fussy. Remember, they're there, they're waiting to get the antigen presented to them. Okay, so the perfect helper T cell with the right receptor is waiting for an antigen presenting cell, like a dendritic cell. And yeah, and then there has to be the handshake and the cytokine signal, the conversation, and then it will proliferate, and then some of the offspring may become cytotoxic T cells. Okay. What has to happen to produce antibodies and memory cells? A, plasma cells must interact with the antigen. B, helper T cells must have a handshake and send a signal to the B cell. C, B cells must come in contact with cytotoxic T cells. D, plasma cells must phagocytize and present the antigen. E, helper T cells must have a handshake and a signal with the natural killer cell. Okay, so we're making antibodies here. Think about it. Yeah, that's the B cell line. That's the humoral immune response. Now they can make antibodies on their own, but we're trying to, okay, we're trying to make memory, right? So what has to happen? Right, you have to have the T cell there. So the helper T cell must be involved, have a handshake, and send a signal to the B cell. So it's B. Okay, right. B is correct. So the helper T cell must be involved. Which part of immunity would not act best against free-floating viruses? Or excuse me, which part of immunity would act best against viruses free-floating that have not invaded a cell yet? So they're just like any other particle bouncing along. So cell-mediated immune response, by definition, humoral immune response, natural killer response, cell-mediated immune response, and natural killer response, humoral and natural killer cell response. Well, of course, the humoral response. Okay, so the cell mediated, really, the T cells are all about going after the virus if it's invaded one of our cells, if it's turned a cell against us. But no, if it's free floating, it's humoral. And then we make antibodies. The antibodies will label the viruses for destruction and it will neutralize them so they can't even attach to the host cell. Okay. So cytotoxic T cells and natural killer cells, they also only go after cells that have been infected. All right, so hopefully you're listening. Which cells would directly attack an infected cell that no longer looks like self? It is betraying us and is displaying foreign epitopes. Oh no, foreign ID tags. Helper T cells, B lymphocyte, natural killer cells and B cells, cytotoxic T cells and natural killer cells, cytotoxic T cells and B cells. Now you got to think about this one. So they're already in the cell. Now remember, the CD8s would take out this particular cell that's been infected, right? So natural killer cell and cytotoxic T cell and natural killer cells. It would be D. They are the CD8s. That's what they do. Kissing cousins, those two. Look alike, act alike. But the cytotoxic T cell is specific. Okay. Which specific cell involved in immunity can recognize a sugary bacterial capsule and has to work alone in this? B cell, helper T cell, cytotoxic T cell, natural killer cell, erythrocyte. Yeah, little B cell alone, no memory. Which defenses would act best against altered cells that have become cancerous? Cell-mediated, humoral, innate, cell-mediated immune response and natural killer cell response, humoral and natural killer cell response. Okay, so the cell-mediated immune response, but let's move on. Humoral is extracellular stuff floating around. Innate, no, that's just kind of barrier stuff, so no. So it's got to be, again, cell-mediated immune response and natural killer cell response. D. Which cell is the actual antibody-producing cell, full of antibody factories? Plasma cells, T lymphocytes, B lymphocytes, macrophages, natural killer cells. It is the plasma cell. Right. Plasma cells came from its parent is the B lymphocyte, but the B lymphocytes divide, make baby cells. Some of them are plasma, some are memory. Okay, it's the plasma cell. This cell can destroy altered non-self cells. It kills by punching a hole in the non-self cells. It is not part of specific immunity. Okay? Magic Johnson owes a lot to this. Okay? That should a lot to this. Okay. Helper T cell, macrophage, natural killer cell, B lymphocyte, suppressor T cell. Okay, it's got to be the natural killer cell, right? It wouldn't even be the cytotoxic T cell because it's not part of specific immunity. So natural killer cell. C. Okay. If you were the recipient of a new liver or liver transplant, which type of immunity would give you the most concern? Cell-mediated, humoral, lysozyme, cytokine, political. I know. It's hard to come up with names. Cell-mediated. A is correct. Good. This immunoglobulin is found in greatest numbers in mucosal membranes and would protect you best from dental disease in the mouth and where there's a lot of mucus and sexually transmitted infectious diseases. IgA, IgG, IgM, IgE, IgD. You have to think about it. So mucosal membranes, it's got to be IgA. IgA. And they do. They protect you from dental disease. When you get stressed out, your IgA levels drop and you get more dental disease, by the way. So go to your dentist. Until then, clean your teeth. Now, here's a thought. Is it possible for us to make memory cells, like memory B cells, without disease? I don't know. Could we I don't know, introduce like the bits and pieces of the antigen and maybe from that make a memory cell and get that so that we wouldn't have inflammation and symptoms? Yeah, you can. It's called a vaccine. Okay, so let's talk about vaccines for a moment. So we're going to switch gears. And if you want to take a break, that's fine. Stretch. Okay, take a break or not. Okay, so what is the purpose of a vaccine? Well, as mentioned earlier, the whole idea is the introduction of antigens to protect animals from infection, make memory lymphocytes, hopefully without disease. So vaccines, so what have you heard? A lot, I'm sure. We've all had an earful. Well, when responding to some conversations, it's very important to have your facts. How do you get your facts? Well, you now know how the immune system works. So you have a lot of information, a good foundation from which to build. Let's talk about the vaccines. And I'm going to talk about the good and the bad. Okay? Vaccines have had problems if you don't treat them properly. Yeah, this is not good. Your immune system is very powerful. Look at what a super antigen can do. And you have those cytokine storms. Okay? So this is, you got to be really important. You got to be really careful. So let's understand vaccines. First of all, not all vaccines are manufactured the same. If you're thinking there's some government body making vaccines, wrong. They are made by a vaccine manufacturing company, probably one of the big pharmacology or pharmaceutical companies. They're manufactured, hopefully according to the directions of scientists. Now, not all pathogens are the same. Remember what I said? What has to happen to make memory cells? We don't respond to everything the same way. Okay? Some are intracellular, some are extracellular, some have different kinds of epitopes, different chemicals, proteins, lipids, carbs. Not all people are the same. Oh. So are you going to make the memory? If I give you the vaccine, are you even going to make the memory cells? Well, I don't know. Ask your parents or ask your genetic makeup, ask your genome. So let's go into the vaccine types. Again, not all vaccines are the same. One type of vaccine is called the attenuated vaccine. This is where you have the whole microorganism. It is alive, but it is weak. They changed it in some way. Can't cause disease in someone who has a healthy immune system. Okay? If somebody has a healthy immune system. Okay. We could also call it active. Why use this one? Well, it best mimics an infection. The attenuated ones tend to be intracellular pathogens. So if they're living, they can get inside our cells and replicate, but not very well. And that way you can bring in the T lymphocytes. If you can bring in the T lymphocytes, you can make memory. Okay? So there you go. You can get memory, you can get both memory T and memory B cells. Okay? So it does the best job. Best mimics the infection, producing lots of memory and long immunity. So again, this is important for extracellular invaders because it will give you memory B cells and memory T cells too. Okay? So once they get into the cell, well, okay, then the cell-mediated immune response takes over. You got memory T cells. Wouldn't that be good? Wouldn't that keep it away? Yeah. Then there are the killed or inactivated kinds of vaccines. So to call it killed is kind of a misnomer. We can call it inactivated. They are inactivated with heat or chemicals like formalin. Come here. This is only going to work with extracellular invaders. Okay. So, as far as memory B cells, yeah, that is probably all you're going to get. The if they're extracellular, they're not getting in our cells, so you're not going to get memory T cells probably. So no memory T cells, and you're probably going to need lots of boosters. Memory's not that great. Toxoids. Remember what I said? Sometimes the bacterial cells are scary, but the toxin will kill you immediately. The toxin will attach to important neurons that allow you to breathe. So, yeah, it's the toxins that we're scared of. And a lot of vaccines are not against the bacterial cell and its epitopes, but against the toxin. So, we inactivate the toxin and it is now a toxoid. The toxoid is changed. They heat it, they give it chemicals. So, yeah, so for some microbial infections, the symptoms are due to the toxins. Heat or chemically inactivated. But again, it's only bits and pieces. It doesn't replicate in a cell. So you're only going to get memory B cells and no memory T cells. And you're probably going to need boosters. Okay. Subunits. Well, this is just bits and pieces. Why put the whole organism in there when you just put like little epitopes? So only part of the microbe, something that all, some epitope ID tag that all of those microbes carry. Okay, and we can target that. I don't know what's going on with that. So, but again, it's particles. You're only going to make B cells, no memory T cells. You're going to need boosters. All right, how about a quiz question? Which of the following vaccine types would you develop if you wanted to best mimic the natural progression of a viral disease? Patient would probably not require boosters, at least not for many years. Dead or inactivated, toxoid, attenuated, subunit, or virulent pathogenic. Okay, you don't want the virulent pathogenic form. Yikes. Of course, it would be attenuated, right? They're alive enough to replicate in the cells. You get T cells. And if T cells get involved, you get both memory T cells and you'll make memory B cells. The virus is going to spend some time outside of the cell, so the B cells can label it for destruction with their antibodies. So you get both. Yay. A toxoid vaccine is made of viral particles, is made of inactivated bacterial toxin, is made of a whole toxin producing bacterium, should be bacterium, is made of bacterial capsules, is made of memory T cells against bacterial toxins. B is made of inactivated bacterial toxin. Yep, you got it. A dead inactivated vaccine is changed but weak, living but weak, changed but could revert. B, a vaccine that only contains an antigen fragment. C, a microbe killed with heat or chemicals. D, a vaccine that requires boosters. E, a vaccine that contains preservatives. Okay, so it's killed, a microbe killed with heat or chemicals. Basically inactivated, right? All right. You don't have to memorize all of these. I just want to show you what vaccines typically look like. DPT, diphtheria, pertussis, and tetanus. They tend to be toxoid, diphtheria, toxoid, tetanus, toxoid. And pertussis is a protein, long story there. Haemophilus influenza, HIB, is part of the capsule. We'll talk about that one. Just kind of measles, mumps, rubella. These are active, attenuated. And the chickenpox virus, varicella zoster, is active, but attenuated. Okay? This will, why would they do that? Because again, you get the best memory if they're living, but weakened, right? You get memory B and T. That's good. You get both cell-mediated and humoral. If you've got a big a big force coming at you, an enemy force, don't you want both branches of the military working and remembering and getting better and faster? Yes, but there are consequences sometimes. Okay, so I just wanted to show you, oh gosh, let's not do this. Okay, this is just showing the historical comparisons of morbidity and mortality for vaccine preventable diseases in the United States, according to the Journal of American Medicine. But anyway, this summarizes it, and I can't see this with my eyes, so I have comments relating to that a little bit later on. If you want that journal article, I will send you the PDF, okay? Actually, I might even post it. Okay, so let's talk about what else comes in the manufactured vaccine. Because there's more than the pathogen. Okay, there's always extra chemical components. Cell components, if it's grown in cells, viruses have to be grown in living cells. A lot of viruses, okay, that we make vaccines for, they have to be grown in eggs. So if grown in eggs, you will have egg proteins in the vaccine, which could be a problem if you become allergic. Okay. Formaldehyde in very small doses. That is also used to kill viruses. Preservatives so that you don't have to refrigerate it. It's cheaper. You can ship it all over the world. Stabilizers that include gelatin and other things. Because we don't want cross-contamination if you reuse the same vial over and over. Usually they're antifungals and antibacterials, so you don't get an infection when somebody punches a needle through your first line of defense. Adjuvants and conjugates. I'm going to talk about conjugates in just a moment. Influenza, the flu vaccines, they tend to have thimerosal, but not in the children vaccines, only in the adults. Okay, because mercury is just not something we like. And I'm not crazy about it, and I'm glad a lot of parents got on the government to say we don't want mercury as a preservative, as an agent to, to make the vaccines anymore. And that's fine. And again, it is, it is in small quantities, but as a very smart student of mine once asked, well, so what did they replace it with? Good question. I don't like thimerosal and I don't like preservatives in general, but I understand their purpose. Okay. Okay. What about this conjugate? Well, here's something else that might be in the vaccine. What if your pathogen that you're trying to make a vaccine against makes a sugary capsule? Remember, immune response doesn't respond to sugars, right? So like bacterial capsules. And because you don't want to die of meningitis or something like that, you would really like some protection, especially for very young children. Because some of the most invasive bacteria have capsules, and they are especially virulent against children under the age of 4. Okay. And so the most virulent types of encapsulated bacteria would be Haemophilus influenza, type B, which we call Hib, Neisseria meningitidis, yeah, causes meningitis, Streptococcus pneumoniae, and there are some others, but these we actually have vaccines for. And but again, why are the children so vulnerable? Well, understand this. This goes back to the antibodies. Do you remember the IgG antibodies will last in mom for about 6 months? Unfortunately, people will be coughing and sneezing these bacteria, and believe me, there are carriers out there, and they feel fine, and they love babies. So now I'm probably making you paranoid, but okay, if everybody has their vaccines updated, we're all okay here. All right. So mom can protect for about 6 months. If you're thinking the colostrum, well, that is for the GI tract. These are respiratory types of pathogens. So infants and young children, how are they going to respond to the capsules of these bacteria? They're not. They're going to fail to mount antibodies to that sugar, and so they're not going to be able to respond and get protection. Okay. So children are very susceptible to these infectious agents and they get very ill. So the solution, attach the carbohydrate to a protein. So if there's a capsule, a sugar capsule, staple on a little protein, like an extra tag on it. Believe it or not, that works, and that's an adjuvant. So that actually works. So, okay. Once again, I just wanted to point out vaccines are manufactured by drug companies, and there are only a few out there. But I also wanted you to know that this is a commodity. It's sold. Hopefully there's enough government regulation and they're being monitored well enough. But there are many companies. So if you think you only have the vaccine that the doctor gives you, there might be something else available. Okay. So again, how are vaccines made? It just depends. Who makes them? It depends. So I just wanted to show you the influenza vaccines. Look at all of these, and these are the companies that make them. You probably aren't familiar with many of these, maybe GlaxoSmithKline, they're really big. They make a lot of antibiotics. But all of these are producers of influenza vaccines. Okay? Okay. And as far as the Neisseria meningitidis that causes meningococcal disease, here are the different companies that make vaccines against that. So you sometimes have the opportunity to shop around. Unfortunately, this is going to require a lot of work on your part, but if you don't like a particular vaccine, you might be able to look around. Just saying. Oh, I'm sorry, that is a startler, but I had to do this because I needed to make a point because there are some bad aspects of vaccines that have been used incorrectly, to say the least. And there are on every vaccine. It's never hidden. There's a huge list of all the side effects of vaccines. Do you ever listen to the side effects of all the drugs that they are, pushing on the television? Sometimes I just freak out as if the public really is buying this like a new, like a new kind of, hair product or something. But vaccines can cause problems. How does any scientist know how your body is going to respond to any antigen? Think peanut allergies, peanut proteins. People can go into anaphylaxis because they encountered a peanut. Okay, so it's really hard. They try to think of the best vaccine, the safest vaccine, the one that's going to give you the best memory. Okay, so, and like I said, I'm going to go both sides. I'm going to show you the good and the bad. And, I am pro vaccine, but I respect vaccines very much. Okay, and I don't treat them lightly. Again, vaccines extremely powerful tools because they manipulate your immune system. Okay, they get your immune system to form an inflammatory response so you can make memory cells. So you're going to have symptoms. Okay, maybe mild, maybe severe, but it's tricking your immune system to go into inflammation mode, battle mode, and you can get symptoms. So the immune system is designed to save, but it can also kill. Think anaphylaxis due to a peanut. Vaccines, think the analogy like an automobile. I know that's a very lame analogy, but you know, automobiles, we love them. We know that it really helps us on a daily basis. And it's important, to manufacture and use them properly. If not, they're very deadly, right? I know it's a different thing, but it's close. So let's begin. If you're just horrified by this image here, believe it or not, this man did survive smallpox. The toxemia should have probably have done him in. He is in bad shape. Um, we have had a vaccine against smallpox. It's eradicated. That's a nice thing. You don't have to worry about whether or not you like the smallpox vaccine. Okay? It's gone. You don't need it. You didn't have it. I had it because I'm young enough. So these are some of the statistics that I took from that one chart that you can't read, from that journal that I can give you. We used to see about 29,000 cases in the US each year of smallpox, and about on average, and about 337 deaths. Now, I'm not showing you the peaks here. So, taking these numbers from 1900 to about 1949, okay? So, on average, this is how many cases we saw. So, imagine if this was a loved one, you would not be very happy about this. We don't worry about smallpox and because we don't see this, right? Thank goodness. Okay, now understand though, the vaccine is nasty. The vaccine was nasty. I had it and I'd have it again if really smallpox was raging through the community. However, it is active, it is attenuated, and it is called the variola virus vaccine. And yeah, really, really some severe problems. So, this is what can happen. It's really important when military people got this when everyone was freaking out after 9/11 and they thought, well, let's just give everybody a smallpox vaccine. Why not? And I just couldn't believe it. If you're going to go after, if you're going to scare people into thinking there's a bio threat, why choose smallpox? Why choose that vaccine to try to make the public feel better? Because it is bad. And, there are some cases where people would scratch the lesion, they get the viruses on their fingers, and they would rub their eyes, they would spread it to their family. It is not a pretty kind of vaccine. It's better to get it when you're a child, certainly than an older person, but this is bad. Okay. So this is called progressive vaccinia. It's a lesion. Again, it was from somebody in the military. Ugh. But again, we don't want smallpox. Good, it's gone. We don't have to worry about it. Yay, eradication. Okay, so how about polio? Polio is gone out of the United States. It's nearly eradicated off the planet like smallpox, thanks to universal precautions. So again, we used to see epidemics resulting in about 13,000 to 20,000 paralytic cases per year in the U.S. Now remember that if you're paralyzed, that doesn't go away. you're paralyzed. And then the next year you get the next group, okay? And then you're all paralyzed. So this is called poliomyelitis. You get the virus, it produces paralysis, and this was horrible. The viruses were spread throughout the United States, and you had all these young children. If they were, if they were lucky, they didn't get a symptom or they just got GI symptoms. Unlucky, they were on crutches. Very unlucky, they ended up in an iron lung because, well, the neurons that run the diaphragm aren't working anymore. There are people that grew up, they are still alive today, that got polio as a child, poliomyelitis, and they've had to live in an iron lung all this time. So again, horrible. Now there was a scientist. Oh, and I should also mention, sorry, I should have mentioned that my aunt, she used to go swimming in South Omaha, Bellevue area, and she got poliomyelitis and she spent her life on crutches and then later on in a wheelchair. So this was nasty. There was one scientist, Jonas Salk, that worked tirelessly. He wasn't a very sociable person. He was kind of strange, but he was horrified by the number of children paralyzed. So he worked so hard, and he came up with an attenuated vaccine, and it worked. And he said, "Okay, so let's make this vaccine for everybody." And he gave out the instructions, the strict instructions to the vaccine manufacturer, and it's going great until one of the manufacturers called the Cutter manufacturing pharmaceutical company made a poliomyelitis vaccine, see Cutter, and this was bad. So this was April 1955. About 200,000 US children got this polio vaccine. And the way the viruses were made, they absolutely had to be inactivated. Okay, they had to be killed. And the cutter didn't follow the directions. They diluted everything, cut a few corners. So the children were getting virulent viruses. So they didn't, and Salk gave them instructions and said, don't do this. So 40,000 children developed polio. So why did this happen? Jonas Salk was working very hard. He knew what had to be done, but government regulation, okay, it wasn't there. You need to have the government breathing, over their necks, and sometimes they're just not always there. So. Okay, so that's the history, a little bit of history, except Jonas Salk's vaccine eventually was exchanged. They changed it out for an attenuated polio vaccine. And Albert Sabin vaccine, he invented the attenuated type. These two men really despised each other historically. It's interesting. They both had, public health in mind. I mean, they really wanted to save lives and make lives better. But Albert Sabin knew that his attenuated polio virus would be better. But Jonas Salk thought, oh my gosh, how could you do that? They're living viruses. But okay, why would we go today to the Sabin vaccine? We're using one of his types or a form of his. Remember, if it's attenuated, if you do it right, if you really attenuate it well enough, which wasn't done at the Cutter factory, then you get your helper T cells, or excuse me, you get your memory T cells, you get your memory B cells. You get more memory and more branches of the military. Okay. Let's talk about the HPV, human papillomavirus, which, by the way, is inactivated or dead. Okay, it's not going to revert. And by the way, this is not just an antiviral, but an anti-cancer vaccine. So with this, it prevents oropharyngeal cancer and cervical cancer. Okay, so it's actually a pretty darn good vaccine. And if you go online, you will find everybody talking about some cases. You have to look a little deeper, okay? You'll find out that there were extenuating circumstances with that, that person who had HPV or that person. I could have prepared that, and I'm trying to remember the case. I'm sorry. Maybe if I can remember, I'll give it to you as an appendage. Okay, so all right. Let's talk about whooping cough. Okay, why? Because Bordetella pertussis is the bacterium that causes whooping cough. Pertussis is part of the DPT vaccine. I wanted to just mention how important it is to not have this in your community. So this is a gram-negative bacterium, so it has LPS, okay, an endotoxin, can cause shock and a lot of other problems. It can also make other toxins in addition to LPS. It is a respiratory bacterium, which means that you can probably spread it a day before you have a symptom, and people do. They get kind of cold symptoms, and they go out, they think they're fine, and they give whooping cough or pertussis to everyone around them. And some people are carriers of pertussis. So it enters the respiratory tract. It kills the cells because well, I should say the bacterial cells will die and then they release the toxin, releasing LPS, that endotoxin we talked about. Again, some people can be asymptomatic, some people can have substantial symptoms. So, okay, this is in adults, but what about children? It can be lethal in children. So if you're not into getting a booster or you know somebody who, I don't, who gets whooping cough? I haven't had whooping cough in years, I'm fine. Well, okay, what if they're an asymptomatic carrier and they want to go play with the little kids? All right, they need a vaccine. If you remember, I showed you this picture earlier. Here's Bordetella pertussis, the bacterium, and it's clear cutting the cilia, and it will cause a very nasty coughing, progression of coughing that can lead to even suffocation. Now, the reason I bring this one up, and I'm not going to bring the diphtheria or tetanus, but I bring this up is because the DPT original vaccine was not a really great one. But now we have the DP or DTAP, DTAP. It's much safer. So again, it's still the acellular pertussis part. The P is really important. This is the pertussis, the whooping cough antigen that we're trying to introduce. So again, the DPT had problems. Now, why is this? This vaccine, the original DPT pertussis, was the whole bacterial cell. They left the endotoxin in there. The LPS was in there. Yeah, so you injected everything, LPS and all, into the patient. Yeah, I don't want to say duh, but duh. Okay. And I suppose they couldn't do anything. I mean, it was probably the only way to do it to make the vaccine work. But they have new genetically engineered non-cellular preps, so you don't have to have the whole bacterial cell in the vaccine. So that's the acellular pertussis, no LPS. It's pretty darn good in that you don't get the symptoms you used to and the bad inflammatory response. But, well, to be honest, if you had the whole bacterial cell, you made more different kinds of memory cells. So not everybody that gets it seroconverts, and the memory cells don't last long. So, because if you do the whole bacterial cell, you have all of those epitopes, and you can actually make all kinds of memory cells against all of the epitopes. So yeah. So when the vaccines don't work, you send in the poisons, the antibiotics. Okay. So you give it to people if you've been exposed. You get the antibiotic even if you've had the vaccine recently, and you even give it to people who have the full-blown whooping cough. Okay, so measles, the reason I'm talking about measles is because this is the next to be eradicated. I mean, it's hard to design a vaccine that we can actually use again. I mean, there's a lot of infectious organisms, but like I said, there are problems. Sometimes we can't make safe vaccines. Sometimes we can't make vaccines that give lifelong protective immunity. There's other problems you have to be able to the pathogen can only be in the human population. If it can spread to animals, the vaccine won't work, but our universal vaccine won't work for universal eradication. But measles, okay, so we've gotten rid of smallpox. We're about ready to get rid of polio. Measles will be next, we hope. So, little bit about measles. Measles is whenever you get a vaccine for measles, it's the M and measles, mumps, and rubella vaccines, okay? But why do we want the measles vaccine? Well, because before we had vaccines, we had about 530,000 cases every year in the United States on average. Again, that doesn't include outbreaks, big peak seasons, okay? Now, of that, we had about 440 deaths every year in the US. So let's go into what measles is and what it does. Measles is viral, and it's respiratory, so it's spread very easily from child to child and adult to child. It causes all respiratory symptoms because it's a respiratory virus, cough, runny nose. Because it's a virus, they have a tendency to cause rashes. This one has a unique rash that starts at the hairline and moves downward. Also, under the little tongue and in the mouth, you might see Koplik spots, little white spots, and a high fever of about 104 degrees Fahrenheit. Not good. So the child is sick and miserable and inconsolable. But there are complications. Sometimes these viruses cause more problems. They can cause ear infections, get into the lungs, and cause pneumonia. They can get into the central nervous system and cause encephalitis of the brain tissue. And that happens in about 1 in 1,000 cases. So think about all of these cases, all right? The 530,000. So, 1 in 1,000 got encephalitis on average. And from that, you can get death or permanent brain damage. Um, Speaking of brain damage, sometimes it causes something called subacute sclerosing panencephalitis. That occurs in 1 in 100,000 children who get measles. That produces permanent dementia. So your child is going to have dementia. Think about elderly with severe cases of Alzheimer's and dementia. And there's no cure for this. There's nothing you can do. Um, and also understand that measles will immunosuppress your child and all of your patients. Measles causes transient severe immunosuppression. So, you know how AIDS makes you depressed? Yeah. So, so will measles. A lot of viruses do this. And if that happens, you are prone to secondary infections. So, of all these children and people and adults who got measles, okay, so how many went on to get ill from something else and perhaps die because they were immunosuppressed? Okay. And the big problem is pneumonia. So other things come in and infect the lungs, and you're too weak to do anything about it. Okay. And the pneumonia is the main cause of death. Measles outbreak. Yeah. We had it in Disneyland, I think. Yeah. Um, a lot of people do count on something called herd immunity. So let everybody else get the vaccine. I won't. Understand because everybody's a little different genetically, some people do not seroconvert, meaning that they don't make enough memory cells. They don't make enough antibodies. They don't make memory cells, so they're not protected. You probably know a lot of people that went in and got their plasma checked and they had to go in and get more vaccines, more boosters because they didn't seroconvert. Well, a lot of children and adults, they don't either. It depends on your genes. So, and there are a lot of people out there that can't have the vaccine, any particular vaccine, for whatever reason. They react badly, can't have anything with chicken proteins in it, what have you. There are people out there we do not give these vaccines to, period. So we rely on herd immunity for that. What does that mean? That means that you have to make sure that enough people around you are either completely healthy or immunized. But if you add one person who is sick and not immunized, Okay, well, this isn't going to work. And on average, the people who surround you must be protected by memory at about a rate of 95%. So everybody who you come into contact with, okay, 90% of them have to have coverage for that particular vaccine. But all right, you get together and we're a global community. You might have people who let their vaccines lapse. So they're not making memory cells, they're not protected, they're carriers of what have you. So you can't rely on herd immunity. You can't. We want to leave herd immunity for those people who are very sick or for the very young. Okay? All right. Maybe you've heard this. Measles, mumps, and rubella vaccines cause autism. Okay. Well, there was so much press. This was really big about 16, 17 years ago. And it was all over the press, and everybody was nagging the politicians. And the medical community said, okay, so there was an Institute of Medicine report that came out and said, okay, we've looked at all the evidence, everybody who's done research, and we have to reject any relationship, a causal relationship on a population between measles, mumps, and rubella vaccines and the autistic spectrum disorder. And also they couldn't even find any proven biological mechanisms that would explain the relationship. So not only do you not have any evidence, but we can't even really figure out for sure why that would occur. Okay? You can hypothesize, and I can hypothesize, but you need evidence. Okay? If you're going to make medical decisions for the whole community, for the planet, you need scientific evidence. And all of this happened because the press got a hold of it. Yes, I know. Yikes. But maybe you heard about this guy. This is Andrew Wakefield. He has a degree. Well, he's an MD actually. And yeah, he led public opinion against the measles, mumps, and rubella vaccine. Now, he was a gastroenterologist in 1998, and he was a lead author in The Lancet, a Lancet article. Now, if you're thinking, what's The Lancet? Okay, The Lancet is like the Super Bowl. If you are a researcher and you want your research published, you want it published in The Lancet. Mind you, everybody was, getting all worked up about vaccines because they were scared. So this was a very popular topic and it was published very quickly. And yeah, so and the study, if you look at the study in the Lancet, it looked at 12 random children. And okay, raise your hand if you've had a statistics class. Anything, even just a little. Can't even remember. Okay. How about taking data? All right. 12, is 12 a good population, kind of a good population testing group, do you think? 12 children? Hmm. Okay. But his work did connect the measles component to autism. Yeah. Oops. Or did it? So, yes, the Lancet was just a little embarrassed, and scientists everywhere were thinking, are you kidding about this? So his license was taken away. He was disgraced. The Lancet should have been really held very much accountable for this. But a little bit more about Andrew Wakefield, former MD. So, yes, that's his article there. He received money from a law firm to make his new vaccine, his own alternative vaccine. And okay, so the law firm that he got the money from was representing patients that were involved in vaccine lawsuits. He personally took out a patent, like I said, for an alternative measles vaccine. Used that money for that. He altered his records. So with 12 children, please, and he still had to alter the records. By the way, the 12 children were referred to him by the said law firm. He did lose his license. One other thing too. Speaking of unethical, he paid children at his son's birthday party to participate in blood drawing for an experiment. This is so unbelievably unethical. There are villains and greedy people and sociopaths everywhere in every field. Unfortunately, they're the loudest. Um, is he really the main villain? I think I could blame the press. I could blame the Lancet. But the problem is that he induced so much fear into parents into everybody who we love children. Everybody loves babies and children. So, okay, even if you heard that this was retracted, wouldn't you just think twice? But maybe, just maybe, he had something there, you know? So that's something to think about. And it's scary. People were frightened to death because of this. Okay, yeah, 12 children, really. The Lancet should be ashamed. But that just shows how human everybody is, including the big important editors and reviewers at the Lancet, because everybody was getting all bent out of shape about vaccines. But you need evidence, okay? And the media is just going to latch on to this. By the way, there are still people who quote Andrew Wakefield. They think that they all ganged up on him. They do, but there's always those. Okay, so according to Time Magazine, a quarter of parents trust celebrities. So here's Jenny McCarthy. Okay, she has a wonderful little son, and her son is on the autistic spectrum disorder. I mean, he's on the spectrum, right? So of course, when this happens, and you're this wonderful celebrity, you're going to be mad, and you need to know. Okay. But why would you trust Jenny McCarthy? Okay, there are immunology experts who went to school and studied chemistry and cell biology and immunology and all of this. And do they still make mistakes? Well, sure, but please. All right. She's not an expert. She's not. She'll certainly look better in a bunny suit than I ever will. Even in my heyday. Yeah, TMI. I don't blame you. Okay, so how about some other studies trying to link autism to the measles, mumps, rubella vaccine? Okay, so let's do a better study. Um, so Denmark did a study between 1991 and 1998, 7 years of work. They looked at 440,000, just over 440,000 vaccinated children. And they had to look hard in Denmark, but they found almost 97,000 unvaccinated children. So, okay, then they would have found the unvaccinated children would have less of an autism rate, right? No difference. Okay, no difference. Yeah, but those Danish, do we really want to trust them? Well, let's keep going. Japan, because of all the press, suspended the measles, mumps, rubella vaccines in 2005. Oh, well then if you looked at the rates, of autism, it should go down, right? No, autism rates continued to climb. Finland. Finland, known for providing the best education in the world. They studied 1.8 million children for 14 years after the measles, mumps, and rubella vaccine. You know where I'm going with this. Yeah, they can't find a link. They just can't. So, what is autism? We know that it's first identified in toddlers between 18 to 30 months of age, and that's just exactly when children get their vaccines. Okay? It coincides with that. So, the cause is unknown, but there is increasing evidence that it is linked to a genetic condition. I listened to one investigator. He said that you can tell while in the womb that the placenta is folding abnormally in a lot of children who are developing autism. Now, that's before they got the MMR vaccine, right? So, are there environmental triggers? You know what? as with most things, like I said, there's always probably a genetic component and an environmental component. But okay, we don't know what it is. We don't have anything, any empirical evidence. That's what scientific method is, right? You go through, you have an experimental group, you have a control group, you have double blind tests, you look at many tests throughout the world, and yeah, we just don't have it. Okay, one last thing, and we're good. The pollen counts are so high that junkies are trying to convert the meth back to Sudafed. I love this. Anyway, I know this was a lot. If you have any questions, just let me know. And again, a lot of this lecture includes the questions. So, yeah, it's sort of if the review session, I could have it separate, but I don't want to do that. I want you to listen to my lectures. So, if you have any questions, let me know. You have a great week.